Hello there, and welcome to the Drift to the Stars. I'm so glad you found your way here tonight. Wherever you are right now, lying down, settling in, letting the day begin its slow retreat, this is a space made entirely for rest. There is nothing to solve here, nothing to finish or remember. Only this, a quiet hour or two, drifting gently through one of the most extraordinary and overlooked kingdoms on Earth. A kingdom that lives beneath your feet, inside the trees, outside your window, in the bread that rose this morning on your counter. Tonight we wander into the world of fungi, ancient, patient, and stranger than almost anything else alive. And just before we begin, if these calm, curious explorations help you unwind, consider liking and subscribing to the channel. It helps others find their way here too, one sleepy soul at a time. Now, if you're ready, let's begin. There is a tree outside somewhere, perhaps one you have walked past 100 times without stopping, and if you could follow its roots downward into the dark, past the topsoil, past the stones, you would find something unexpected waiting there. Threads, pale and fine as spider silk, woven between the root hairs in a net so delicate it seems impossible that anything so slight could matter. But these threads are mycelium, the living body of a fungus, and they are doing something that no plant can do alone. They are reaching further, feeding deeper, sensing more. And here is the thing that tends to settle quietly in people when they first encounter it. At the genetic level, the organism producing those threads is more closely related to you than it is to the tree whose roots it embraces. Fungi share a common ancestor with animals, not with plants. Their cell walls are built from chitin, the same material in the shell of a crab, the wing casing of a beetle. They breathe oxygen and release carbon dioxide, just as your body does right now in the dark. When you imagined the fungal kingdom as something alien and apart, you were almost right. But the distance runs in the opposite direction from what you assumed. The mushroom in the forest is, in the oldest sense, more your cousin than the oak trees. That quiet kinship, written into the chemistry of cells, invisible to the eye, has been there all along, waiting for someone to look closely enough to notice it. Closeness, it turns out, can be measured in more than ancestry. In the Blue Mountains of a state in the American Northwest, there is a forest that appears, from above, to be simply a forest pine trees, meadows, the ordinary texture of wild land. But beneath the soil of nearly 4 square miles of that ground, a single organism is alive. One fungus. A network of honey-colored mycelium that has been expanding outward from a single point for somewhere between 2,000 and 8,000 years. Every mushroom that surfaces from that ground in autumn, and there are thousands of them, is the same individual. The same creature, fruiting in dozens of places simultaneously, the way a single person might exhale in one long, slow breath. To stand in that forest is to stand inside something, not beside it. The trees are not the forest's oldest residents. They are relatively recent arrivals, growing in ground that the fungus prepared long before their seeds fell. Scientists still debate the exact age of this organism. The honest answer is, we are not entirely sure. What we do know is that it is among the largest and oldest living things that has ever been confirmed on this planet. And it spends most of its existence unseen, unhurried, threading quietly through darkness, doing the slow work of being alive on a timescale that makes human history feel brief and tender. Tetsugoingness, of a different kind, lives in the very smallest parts of this kingdom. A single fungal spore, the reproductive unit that a mushroom releases when it is ready to continue, is so vanishingly light that it barely registers as a physical object. If you held one on your fingertip, you would feel nothing, less than nothing. The air currents stirred by your own breathing would be enough to carry it away before you had time to look. And yet, that spore is complete. It carries within its microscopic shell everything needed to become a new organism: genetic information, protective coating, the capacity to wait in dormancy for years if conditions are wrong, and to awaken the moment conditions become right. A single puffball mushroom, the round, white kind you might kick as a child in a meadow. Watching the smoke billow releases a number of these spores so large it exceeds the total count of every human being who has ever lived on Earth. Not the current population, every human, across all of history, released in a single slow exhale of pale dust that drifts on the wind and mostly lands in places where nothing will come of it. Most spores never become anything, but the ones that do carry the entire lineage forward. It is a strategy of generosity, so extreme it looks almost like indifference, and yet it has worked, continuously, for nearly 1 billion years. 1 billion years is a span of time the mind approaches but never fully enters. What it means, practically, is that fungi were here long before almost anything else we would recognize as life, before forests, before flowers, before the first fish crawled onto land and learned to breathe. The fossil record shows fungal filaments preserved in ancient rock, delicate impressions of threads that were threading through soil, when the Earth's continents were still arranged in shapes that have no names we use today. And in those early ages, before trees had yet evolved, the tallest living structures on land were fungi. Organisms called prototaxites rose from the ground in columns that could reach the height of a 2-story building. Not trees, not plants, but vast fungal towers standing in a landscape otherwise low and bare. We do not know exactly what they looked like from the outside. We do not know whether anything ate them, or sheltered beneath them, or simply grew around them, in the way that life tends to fill every available space. What we know is that for millions of years, fungi were the dominant tall things on Earth, the giants. The ones that shaped the land for everything that came later. Standing in a forest today, surrounded by trees, it is worth pausing to remember that the trees arrived second into a world the fungi had already been preparing for an incomprehensible length of time. Preparation, in the fungal world, happens at pressures that seem impossible for something so soft. A mushroom, pushing up through the ground, looks fragile, pale, domed, easily crushed. And yet, the force that drives it upward through soil, through compacted clay, through gravel, is hydraulic. The mushroom draws water into its cells and uses that water as a kind of internal pressure system, expanding outward against resistance with a force that has been measured as sufficient to crack asphalt, not to push through loose soil to fracture pavement. A mushroom growing through a sidewalk is not finding a gap, it is making one. The pressure builds slowly, invisibly, over hours and days, with no urgency and no struggle apparent from the outside. And then, at some quiet moment in the night, the surface gives way and the cap emerges into air. This is not aggression. There is no intent in it. It is simply what happens when water and cell walls and patience work together for long enough. It is, in the most literal sense, the softest possible force achieving something that seems to require hardness. A lesson the mushroom does not know it is teaching, but teaches nonetheless, in every spring sidewalk and cracked stone path where something pale and domed has found its way through. Finding its way is something fungi do through means that continue to puzzle researchers. Within the mycelial network, those vast underground threads connecting mushroom to mushroom, root to root electrical signals travel, not the way electricity travels in a wire, fast and direct, but slowly, rhythmically, in pulses that rise and fall like breathing. Scientists measuring these signals have found patterns in them, variations in frequency and timing that cluster in ways that resemble, at least structurally, the patterns of neural firing in animal brains. Whether this constitutes something like communication, or something like information processing, or simply a mechanical consequence of fluid moving through biological tubes, that question is genuinely open. Researchers are careful not to overstate what they have found. But what they have found is this: the network is not passive. it responds. When one part of the mycelium encounters a new food source, signals propagate outward through the threads. When part of the network is damaged, the surrounding filaments adjust. The system behaves as though it is integrating information across its entire span, not thinking in any way we would recognize, but doing something that thinking, in its earliest evolutionary forms, might have grown from. The soil beneath a forest is threaded with something that processes the world, slowly, quietly, without a brain, without eyes, without any of the apparatus we associate with awareness, and yet, somehow, not entirely without response. Response to conditions we would consider lethal is something certain fungi have refined into an art form. In 1986, after the reactor at Chernobyl failed and the surrounding land became one of the most radioactive environments on Earth, scientists studying the exclusion zone made an unexpected observation. Fungi were not absent from the ruins. They were present in unusual abundance. Certain species, including some that produce dark melanin pigments, the same compound that gives human skin its color in sunlight, appeared to be growing toward the radioactive core rather than away from it. Laboratory experiments that followed suggested something that still sits at the edge of what biology confidently allows, that these fungi may be using ionizing radiation as an energy source, converting what damages most organisms into fuel for their own growth, in a process loosely analogous to photosynthesis but running on radiation instead of light. Scientists are careful here. The mechanism is not fully understood. The evidence is suggestive rather than conclusive. But the fungi are real, and their presence in the ruins is real, and the direction of their growth toward the source of radiation, rather than away from it, is real. In the most contaminated place humans have created, something ancient and patient found a way not merely to survive, but to lean in. Leaning in is not how rain on dry earth works, that smell rises outward, upward, into the open air. You know the scent. It arrives a moment before the first drop falls, or immediately after, when the ground that has been waiting for days finally receives what it needed. That fragrance petrica, from the Greek for stone, and the fluid that flows in the veins of gods, is partly the product of a compound called geosmin, released by soil-dwelling bacteria and fungi as moisture arrives. The human nose can detect geosmin at concentrations so low they are measured in parts per trillion. This is not an accident of biology. Evolutionary biologists suspect we developed this sensitivity over millions of years because the smell of rain on Earth was survival information. It told our ancestors that water was near, that the land was about to become generous. What this means, held gently, is that one of the most comforting smells available to a human being, the smell that arrives with rain, the smell that feels like relief, is partly a signal sent by fungi, rising from the ground into the air, carrying information you were shaped to receive, connecting a sensation of deep comfort in your body to something threading quietly through the soil beneath every landscape you have ever loved. Every landscape holds more than it shows. A puffball mushroom, sitting in a meadow in late summer, looks like a simple object, round, white, the size of a fist or a head of lettuce, sitting quietly on the grass. It gives almost nothing away. There is no visible activity, no motion, no sign of what is happening inside the tightly sealed sphere of its body. But inside that sphere, in the days before it opens, a transformation is completing itself. The interior is becoming spore. Almost every cell of the puffball's interior converts into a fine, dry powder, and when the outer skin finally gives to a raindrop, to a paw step, to a hand that cannot resist, pressing it, what emerges is a cloud so dense it looks like smoke. That cloud carries within it a number of individual spores that exceeds, by a wide margin, the total number of human beings who have ever drawn breath on this planet. Each spore is a beginning that may never begin. Most will drift and land and find nothing hospitable and simply wait and eventually cease. But the mathematics of the strategy is patient and ancient. Release enough, and some will find the right patch of soil, the right moisture, the right dark, and the lineage will continue. The puffball does not know any of this. It simply opens, when the moment is right, and lets everything go. Letting go of one season to wait for another is something certain fungi practice with a patience that reshapes how the word waiting feels. In the weeks after a forest fire, when the ground is black, the air still carries the memory of smoke, and almost nothing appears to be alive. A particular kind of mushroom begins to emerge from the ash. Morels, with their honeycomb caps and hollow stems, fruit in burned landscapes in numbers they rarely achieve anywhere else. They were there before the fire. Their mycelium was in the soil, dormant or quietly active, connected to the roots of trees that no longer exist. The fire did not create them, but something in the combination of charred soil and changed chemistry and absence of competing vegetation sent a signal through the underground network, a signal that whatever those threads had been waiting for had finally arrived. They had been waiting, in some cases, for years. In some locations, decades. The precise trigger remains an area of active research. What is understood is that certain fungi are oriented not toward stable conditions, but toward disruption, not toward the comfortable middle of things, but toward the moment after catastrophe, when the ground is open and the competition is gone and something new is possible. They are not survivors of fire. They are, in a quiet and precise way, its intended inheritors. Among the most ordinary-looking things in any forest is also among the most dangerous. A pale cap, a white stem, gills the color of old paper. The death cap mushroom carries nothing in its appearance that the eye would catch as warning no dramatic color, no unusual shape, no smell that signals harm. What it carries instead is a toxin called amatoxin, which moves through the body with a patience that makes it particularly devastating. Symptoms may not surface for a full day after eating. By the time the body begins to signal distress, the liver has already absorbed damage that cannot be reversed. No antidote exists. No compound undoes what the toxin has begun. Hospitals can provide support, and sometimes a transplant arrives in time, but the mushroom holds nothing in reserve for those who have encountered it. This fact does not sit easily, but it belongs here because the death cap is not an aberration in the fungal kingdom. It is an expression of how completely fungi operate outside human categories of intention. The mushroom did not evolve to threaten. It evolved a defense so effective it requires no advertisement, no warning coloration, no drama of any kind. It simply continues being what it is, in the quiet of the forest, indifferent to the difference between those who walk past and those who do not. Somewhere in the same forest, on the bark of a beech or oak, something white and cascading grows dense, soft, resembling a frozen waterfall or a cluster of winter icicles hanging from the wood. Lion's mane mushroom looks unlike almost anything else in the fungal kingdom, and what it carries inside is equally singular. Two families of compounds, hericenones and erinacines, have been shown in laboratory studies and animal models to stimulate the production of nerve growth factor, a protein the brain uses to maintain and repair its neurons. The research in humans is still early, still appropriately cautious, still working toward the kind of certainty that takes years of careful study to establish. Scientists are honest about what they do not yet know, but the direction of the findings is consistent, and the consistency sustains serious attention. What this means, held gently, is that a mushroom growing from the side of a tree in a temperate woodland produces, as part of its ordinary chemistry, molecules that encourage the most intricate structure in the known universe to renew itself. Lion's mane did not evolve these compounds for us. We simply happen to be able to receive what they offer. The forest was not waiting to heal us, but it turns out, in certain soft and unexpected corners, it can. At the edge of a meadow after rain, in the low morning light when the grass still holds the night's moisture, you might find a circle. A ring of mushrooms, spaced with an evenness that seems too precise for accident, curving away in both directions until it closes on itself somewhere in the grass. Fairy rings have gathered explanations across centuries: spirits dancing, lightning striking, something moving through the dark. The actual mechanism is quieter and, in its own way, more worth knowing. A single fungal organism begins expanding from a central point, pushing its mycelium outward in all directions with perfect equality. As the network grows, it exhausts the nutrients beneath its oldest ground and concentrates its activity at the advancing edge. And it is only at that edge, where the soil is still fresh, that mushrooms fruit. The ring is not a shape the fungus chooses. It is the shape of growth itself, of even expansion made visible at the boundary between the spent and the new. Some of these rings have been aged, by measuring their diameter against known growth rates, to several hundred years. The center looks like ordinary meadow, but beneath that ordinary grass, something has been expanding outward since before anyone now living was born, marking each season's progress in a circle that quietly widens year by year, without witness or ceremony. Without witness, fungi reshaped the entire surface of the land. The ground beneath every forest and field and garden that has ever existed was not always ground. It was rock-bare mineral surface containing nothing that roots could access, nothing that water could be held within, nothing that we would recognize as soil. Something had to work the rock into something else before plants could arrive. Fungi arrived first, alongside early algae, secreting acids that dissolved mineral surfaces and releasing phosphorus and calcium from stone. The organic material left behind by dead fungal bodies accumulated in the cracks, layer by invisible layer, across spans of time that make human history seem like an afternoon. What accumulated became the first soil, not rich, not deep, but enough. Enough for the earliest land plants to take hold in, and from that beginning, over millions of further years, the forests grew. Every gardener who has ever pushed a hand into dark earth and felt its texture yielding, alive, faintly warm, has been touching the accumulated residue of fungal work that began before animals existed. The ground is not a backdrop to the story of life on land. It is something fungi made, slowly, from stone, in the dark, without any awareness of what would eventually grow in what they left behind. What grew in that ground eventually developed a relationship with fungi so deep, it reshaped how both organisms function. Mycelial networks explore soil in threads far finer than any root reaching phosphorus, locked in pores too small for roots to enter, drawing water from distances the plant itself cannot access. In exchange, the plant sends sugars downward through its roots into the mycelium, feeding the fungus with the products of its photosynthesis. This exchange is not symbolic or occasional. It is continuous, chemically regulated, and so fundamental to how most plants function that many species cannot survive without it. Researchers have found that trees connected through shared mycelial networks exchange not just nutrients, but signals. When one tree is stressed by drought or pest, compounds travel through the fungal threads and neighboring trees adjust their chemistry in response. The network is not neutral infrastructure. It is an active participant in the conversation between trees, routing resources, carrying information, shaping the health of the forest as a whole. A forest is not a collection of individual trees competing for the same resources. It is, in significant part, a community held together by fungi, and the fungi are not servants of that community. They are among its most essential members. Among the most essential and least visible parts of the fungal body is the hypha, the single thread that is the basic unit of mycelium. One hypha is so fine that it passes through the spaces between individual soil grains without displacing them. It navigates the microscopic architecture of the ground, the way water finds the path of least resistance, not by force, but by following the geometry of what is already there. And yet from these individual threads, gathered in their millions and billions, the mycelial network emerges with its capacity to cover acres, to persist for thousands of years, to route nutrients across distances that would challenge a root system many times its apparent size. The relationship between the thread and the network is one of the quieter mathematical wonders of the living world. Each hypha operates according to simple local rules: grow toward nutrients, retreat from toxins, fuse with compatible threads nearby, and from those simple rules, across the entire network, complex behavior emerges. No central coordination, no governing structure, just threads following gradients. And from that following, something vast and responsive and ancient taking shape in the dark. The forest floor is woven from a logic so distributed it has no single location. It is everywhere the threads are, which is nearly everywhere. Nearly everywhere fungi have been found. They have also been found doing something unexpected. In the exclusion zone around Chernobyl, one of the most radioactive landscapes on Earth, fungi did not retreat. Certain species were found growing toward the reactor rather than away from it, their dark melanin pigments absorbing ionizing radiation, in a process that may, researchers cautiously suggest, generate usable energy. The mechanism is not fully understood. The evidence points in a consistent direction, without yet arriving at certainty. What is established is that these fungi are thriving in conditions that damage the DNA of most organisms within hours, and that their melanin appears to be doing something more than passive protection. They are not merely surviving the radiation, they may be using it. This sits at the edge of what biology currently allows us to say with confidence, and scientists name that edge honestly. But the fungi themselves are not at the edge of anything. They are simply growing, as fungi grow, into whatever gradient of available energy the environment presents, indifferent to whether that energy comes from decaying wood or decaying atoms, from the ordinary chemistry of soil or the extraordinary chemistry of a ruined reactor. Availability is what matters. Source is secondary. Secondary to the nose, the source of petrichor barely matters. The smell arrives, and something in the body relaxes before the mind has caught up. That fragrance, rising from dry ground at the first touch of rain, comes partly from a compound called geosmin, released by soil bacteria and fungi as moisture reaches them. The human nose detects geosmin at concentrations measured in parts per trillion, a sensitivity so extreme it places this among the smells we are best equipped to perceive of anything in the natural world. Researchers believe this sensitivity developed over millions of years because the smell of rain on Earth carried survival information. It meant water was near. It meant the land was about to become generous. What this means now, held quietly, is that one of the most comforting sensory experiences available to a human body, that smell that arrives with rain, that smell that feels, in some way that bypasses explanation, like relief, is partly a signal produced by fungi, rising from the ground into the air, shaped by evolution into something your nervous system was built to receive as good news. The fungi are not sending it to you, but you were shaped, across deep time, to feel it as comfort. The connection between your sense of safety and the chemistry of the soil is older than your species. The soil holds other signals, sent not upward into air but laterally, through threads, to whatever in the network is positioned to receive them. When a mycelial network encounters a source of food, the threads that found it do not simply absorb it in place. The discovery propagates chemical gradients shift, growth patterns adjust, and resources begin moving through the network toward productive regions and away from depleted ones. Researchers observing this process have described it as the network solving a problem, though they are careful to explain that no solving, in any conscious sense, is occurring. What is occurring is optimization, the same kind of optimization that produces efficient road networks and river systems and the branching of blood vessels in a lung. The network finds the best path, not by planning, but by growth, by the accumulated result of millions of local decisions made by individual threads following simple chemical rules. And the shapes that emerge from this process, the branching, the redundancy, the preference for certain angles and junction densities have been measured and found to be genuinely efficient, not approximately efficient, not efficient by accident. Efficient in ways that match the solutions human engineers arrive at deliberately, after calculation, with full awareness of the problem they are trying to solve. Problems the forest solves continuously, without calculation, include the question of what to do with the dead. A fallen tree, massive, dense, its lignin-reinforced wood, engineered by evolution to resist decay, would persist for centuries if only bacteria were available to work on it. Bacteria cannot break down lignin. For the vast majority of Earth's history, neither could anything else. Then fungi evolved the enzyme systems, capable of dismantling lignin's molecular structure, and the carbon cycle changed. Dead wood could now return to soil. The nutrients locked inside a fallen tree could now become available to new growth. During the Carboniferous period, before fungi developed full ligninase capability, dead plant material accumulated in layers hundreds of feet deep, compressed over millions of years into what we now extract as coal. The coal seams running beneath entire continents are, in a precise geological sense, the record of an era before fungi could fully do their work. When fungi caught up, the accumulation stopped. The dead began returning to the living at a rate that balanced the system. Every forest that has ever renewed itself after a fallen tree, every meadow that has grown from the decay of last year's growth, exists because fungi developed the capacity to do something no other organism could manage. They did not save the carbon cycle. They simply evolved, and the carbon cycle adjusted around what they became. At the edge of where tree roots end, something else begins. The finest root hair of a mature oak extends perhaps a millimeter beyond the root itself, a thread-like projection, feeling outward into soil for whatever it can reach. But wrapped around that root hair, fused so intimately the boundary between them is difficult to see. Even under magnification, fungal threads continue outward into soil the root will never enter. They reach phosphorus locked in pores too narrow for any root. They access water held in distances the tree cannot span, and they carry what they find back to the root, trading it for sugars the tree produces in its canopy and sends downward through its trunk. This exchange has been operating, in its essential form, for over 400 million years, predating flowers, predating most insects, predating the dinosaurs by a margin that makes those ancient creatures seem recent. The partnership is so old it is written into the genetics of both organisms. Most plants cannot complete their life cycle without it. Remove the fungal partner from the equation, and the plant starves in soil that appears, to every other measure, to be adequate. The soil is adequate, but adequacy, for a plant, requires the threads. What looks like a tree standing alone in a field is never, in the biological sense, alone. It is the visible half of a partnership whose other half is invisible, ancient, and essential. Woven through that same soil, finer than the fungal threads themselves, is something that took researchers by surprise when they first measured it carefully. Electrical signals move through mycelial networks, not the fast, directed signals of a nervous system. Something slower, more rhythmic, traveling in pulses that rise and fall over seconds rather than milliseconds. When scientists recorded these signals and analyzed their patterns, they found variations in frequency and timing that cluster in ways that structurally resemble neural activity in animal brains. Whether this constitutes communication in any meaningful sense, whether information is genuinely being transmitted across the network, remains an open question that researchers approach with appropriate caution. What is not in question is that the signals are there, and that they change in response to what the network encounters. A new food source alters the pattern. Damage to part of the network alters the pattern. The system is not passive. It responds to its circumstances in ways that propagate across distances, that involve the whole network rather than just the affected part. The soil beneath a forest is threaded with something that, at minimum, registers the world and distributes that registration widely. What we call that sensing, responding, something approaching awareness, depends on definitions we are still working out. Working out definitions is also what happens when biologists try to place fungi within the categories life uses to organize itself. Fungi are not plants. They do not photosynthesize. They do not have cell walls made of cellulose. They do not grow toward light as their primary orientation. Fungi are not animals. They do not ingest food, do not move through their environment, do not have nervous systems or muscle tissue. They occupy their own kingdom, a category defined not by what they resemble, but by what they actually are. And what they actually are includes this: they breathe. Oxygen in, carbon dioxide out, in a metabolic exchange that mirrors the one happening in your body right now, in the dark, as your chest rises and falls. The chitin in their cell walls is the same compound that forms the exoskeleton of insects and crabs. Their evolutionary lineage diverged from the lineage that produced animals more recently than either lineage diverged from plants. A mushroom on the forest floor is, by the measure of shared ancestry, closer to you than it is to the fern growing beside it. The kingdom of fungi is not a distant branch of life's tree. It is a neighboring one, close enough that the resemblances, when you know to look for them, begin appearing in unexpected places. Unexpected places are where certain fungi have always chosen to live. Inside rocks, for instance. Endolithic fungi, the name comes from the Greek, for within and stone do not grow on rock surfaces in the way lichens do. They grow inside them, threading through the microscopic pores and fissures of the mineral itself, extracting nutrients from the stone and sheltering within it from temperature swings that would damage exposed tissue. In the dry valleys of Antarctica, where liquid water is almost entirely absent and temperatures fall far below freezing for most of the year, endolithic fungi have been found living within translucent quartz, positioned just deep enough inside the stone to receive filtered light while remaining protected from the lethal desiccation of the open air. They grow at rates so slow that what looks like a smear of pigment on the inside of a rock face may represent decades of activity. Their metabolism runs at the lowest sustainable level, not dormant, but barely more than dormant, conserving everything, requiring almost nothing, persisting in conditions that would end most organisms within hours. To find life inside stone, in Antarctica, surviving on filtered light and mineral traces, is to encounter the outer boundary of what persistence means. These fungi are not thriving in any conventional sense. They are simply, stubbornly, continuously alive across spans of time that reshape what the word ancient means. The honey fungus spreading beneath those Oregon mountains is not the only contender for oldest living organism. Certain lichens fungi that have entered into partnership with photosynthetic algae, forming a composite organism that is neither fully fungal nor fully algal, grow so slowly on rock surfaces that their age must be estimated in centuries rather than years. A lichen the size of a dinner plate on a north-facing cliff in the Arctic may have been growing since before the printing press was invented. Some specimens have been cautiously aged to several thousand years older than most trees, older than most human monuments, older than the written records of most civilizations. They grow a fraction of a millimeter per year or less. They do not appear to be doing much. In summer, they photosynthesize briefly when moisture allows. In winter, they freeze, solid, and wait. They have been freezing and waiting and briefly growing for longer than most of the things humans point to as ancient. A lichen on a gravestone is not decoration. It is a resident one that arrived long after the stone was carved and will almost certainly remain long after the inscription has worn away entirely. Worn away by the same fungal chemistry that built the soil, is the stone of cliffs and boulders and mountain faces across every continent. Fungi release organic acids as metabolic byproducts, and those acids dissolve minerals slowly, incrementally, across timescales that make erosion by wind and water seem hasty. Calcium, phosphorus, magnesium, iron elements, locked inside rock crystal structures, become available to the broader ecosystem through fungal activity. This biomining, as researchers now call it, is not incidental to how ecosystems function. It is foundational. The mineral nutrients cycling through the bodies of every plant and animal in a forest entered that cycle, somewhere in their history, through the dissolving work of fungal acids on stone. Scientists studying this process are now exploring whether it could be directed deliberately, whether specific fungi could be used to extract rare minerals from low-grade ore without the environmental cost of conventional mining. The forest has been doing a version of this for hundreds of millions of years, quietly and without waste, converting what is locked in stone into what is available for life. The question researchers are asking is not whether it works. It demonstrably works. The question is whether we are patient enough to learn from it. Learning from it requires accepting that some fungal strategies are deeply unsettling before they become interesting. Cordyceps fungi, a group of species distributed across tropical and subtropical forests, do not simply grow on or near insects. They grow inside them. A spore lands on an ant and begins to germinate, sending threads through the insect's body that spread without immediately killing the host. Over days, the fungus infiltrates muscle tissue and, through mechanisms researchers are still carefully mapping, begins to influence behavior. The ant climbs. It moves upward from the forest floor to a precise height, specific enough that the fungus appears to have encoded a target elevation into its chemistry, and bites down on a leaf vein with a grip so strong that muscle lock continues even after death. Then the fungal fruiting body emerges from the ant's head, releasing spores over the forest path below, where more ants will walk. This is not predation in any conventional sense. It is something older and stranger, a relationship refined over millions of years of coevolution, until the fungus and the insect are locked together in a dynamic that serves exactly one of them. The forest floor, soft and quiet underfoot, is the stage for dramas this precise and this ancient, playing out below the threshold of what a passing human would notice. Passing below the threshold of notice is precisely what the spore does. Released from a mushroom cap in still air, a spore moves in ways that should not be possible given its size and weight. It is too small to be carried efficiently by diffusion alone, too light to fall in any useful direction. And yet spores travel across forests, across continents, across oceans, between hemispheres. Part of the explanation is a mechanism researchers discovered only recently. Mushrooms generate their own air currents. As water evaporates from the moist surface of the cap and gills, it cools the air immediately surrounding the mushroom, creating a slight temperature difference between that air and the warmer air beyond. That temperature difference drives convection, a gentle upward flow of cooler air that carries spores away from the cap and out into the broader air currents above. The mushroom does not wait for wind. It makes the conditions for its own dispersal through nothing more than the physics of evaporation and the temperature of the surrounding air. A mushroom in still air in a closed room will still move its spores. The mechanism requires no external assistance. It requires only moisture, surface area, and the patient operation of thermodynamics. Thermodynamics operates at a different register inside the mycelial network when it routes nutrients between trees. What flows through the fungal threads connecting the roots of a forest is not random. Researchers tracking the movement of carbon and phosphorus through these networks have found that resources move directionally from trees with surplus to trees in deficit, from the light-rich to the shaded, from the established to the newly germinated. A large, mature tree with full canopy access may send sugars through the network to a seedling growing in deep shade below, where photosynthesis alone cannot sustain growth. The seedling receives what it needs, not from the soil directly but from its neighbor, through an intermediary that takes a share for itself and passes the rest along. Scientists debate how actively the network regulates this flow, whether the fungus is in some sense managing the distribution or simply providing the infrastructure through which plants manage it themselves. The honest answer is that the mechanism is not yet fully understood. What is understood is the outcome. Forests with intact fungal networks have higher seedling survival rates than forests where those networks have been disrupted. The network is not neutral. Something about its presence improves the odds for the most vulnerable members of the community. Among the most vulnerable moments in any organism's existence is the one immediately after reproduction, when what was one becomes many, and the many must find their way without guidance. A mushroom at peak spore release is engaged in an act of extraordinary statistical generosity. The number of spores a single large puffball releases in one event exceeds the total count of every human being who has ever lived, not the current population. Every human, across the full span of recorded and estimated prehistory, released in a single slow exhalation of pale dust, drifting outward on whatever air the mushroom has generated for itself or caught from the world beyond. Most of those spores will land somewhere inhospitable. Most will wait and find nothing and eventually cease. The strategy is not precision. It is abundance, carried to a scale where probability itself becomes reliable. Release enough, into enough directions, over enough seasons, and some fraction will always find the conditions required. The puffball does not calculate this. It does not weigh the odds. It simply opens, at the moment its chemistry signals readiness, and releases everything it has prepared into the open air, an act of total commitment to a future it will never observe, in places it will never reach, in lives it will never know began, beneath contaminated ground soil that human industry has left heavy with lead, mercury, cadmium. The residue of a century of manufacturing certain fungi are doing something that no machine has yet replicated at comparable cost or scale. Their mycelial threads absorb heavy metals from the surrounding soil, drawing toxins into their own tissue and concentrating them there, away from the water table, away from the roots of plants, away from the organisms that would be harmed by contact. The fungus itself becomes the vessel of containment. In experimental plots where this process called mycoremediation has been allowed to proceed, the surrounding soil recovers measurable fertility within seasons. Plants return. Insects return. The ground that was poisoned becomes, incrementally, ground again. The fungi that do this work are not harmed by what they accumulate, at least not in ways that prevent them from continuing. They have enzymes adapted to handle metal ions that would damage most cellular machinery. Whether they evolved these adaptations in response to naturally occurring metal-rich soils, or whether the adaptation serves some other primary purpose, and the metal tolerance is incidental. Researchers are still working that out. What is already clear is the result. In the places humans have most thoroughly damaged, fungi are among the first organisms to begin repair. Not because they were sent there, because that is the direction their chemistry naturally runs. Running in a different direction entirely outward, upward, far beyond the reach of any soil, are the spores that fungi release into the upper atmosphere. Spores have been collected at altitudes where weather systems operate, where commercial aircraft cruise, where the boundary between atmosphere and open sky grows thin. At these heights, individual spores drift in air currents that circle the globe, crossing oceans in days, descending with rainfall onto landscapes thousands of kilometers from where they were released. The protective coating of a fungal spore, layered, chemically complex, evolved to resist desiccation and ultraviolet radiation, makes this journey survivable. A spore released from a meadow mushroom in one country may germinate in soil on another continent months later, having passed through temperature extremes and radiation levels that would damage most biological material. What this means for how we understand ecosystems is still being worked through. Fungal species, once thought to be geographically isolated, have been found on multiple continents, their distribution patterns suggesting atmospheric dispersal rather than gradual range expansion. The atmosphere is not simply above the living world. For fungi, it is a medium of travel, ancient, global, and continuously in use. In use for purposes that extend even beyond Earth's atmosphere, if the evidence from space experiments is taken seriously. Fungal spores, exposed to the conditions of open space vacuum, cosmic radiation, temperature, swings between extremes that no terrestrial environment approaches have survived. Not all of them, and not indefinitely, but enough, and for long enough, to sustain the hypothesis that spores might endure an interplanetary journey if protected by rock or ice. This is the foundation of a theory called panspermia, the idea that life, or the precursors of life, might travel between worlds on meteorites or dust, seeding new planets with organisms that evolved elsewhere. Fungi are not the only candidates for such travel, but their spores are among the most radiation-resistant biological structures known. Researchers who study this are careful to distinguish between what the experiments demonstrate and what they suggest. The experiments demonstrate survival under space conditions for defined periods. They suggest the possibility of longer travel without confirming it. What sits at the center of this, quietly, is the image of a spore, something smaller than a grain of dust, as a vessel capable of carrying life across the distance between worlds. The universe, it turns out, may have always had passengers. Passengers of a different kind move through the mycelial network when a forest is under stress. Chemical signals, compounds that neighboring fungi and plant roots can detect and respond to propagate through the threads when part of the network is damaged or threatened. A section of mycelium severed by a burrowing animal, a root attacked by a pathogen, a tree stressed by drought. Each sends a change rippling outward through the connected network, altering the chemistry of the threads in ways that reach organisms meters or tens of meters away. Those organisms adjust. They shift resources, thicken cell walls, produce compounds that resist the same threat. The network does not plan this response. It does not coordinate it through any central structure. The response emerges from the chemistry of individual threads reacting to local conditions, and those local reactions accumulating into something that looks, from the outside, like a coordinated reply. Ecologists studying forest resilience have found that forests with intact mycelial networks recover from disturbance faster than forests where those networks have been fragmented. The connection itself is part of the defense. The threads do not only carry nutrients, they carry the information that allows a community of organisms to behave in moments of stress, more like a single responsive body than a collection of individuals. Individual threads of mycelium, gathered in sufficient density, produce a material that engineers are only beginning to take seriously. Mycelium composite fungal threads grown through agricultural waste like corn husks or hemp fiber, then dried and heat-treated to halt further growth, produces a substance that is, weight for weight, comparable in compressive strength to concrete and superior to many plastics in its resistance to impact. It is also fully compostable. At the end of its useful life, it returns to soil without leaving residue. Companies are already manufacturing packaging from it, replacing expanded polystyrene in shipping boxes for electronics and fragile goods. Architects are exploring it as insulation and as a structural element in temporary buildings. The material grows into its final shape rather than being cut or molded from a larger piece, which means it produces almost no manufacturing waste. The fungus does the forming. What humans provide is the substrate, the template, and the decision about when to stop the growth. Everything else, the binding, the density, the structural properties, emerges from what mycelium does naturally when it has material to thread through and time to work. The forest has been making this material for hundreds of millions of years. We are approximately 15 years into learning how to use it deliberately. Deliberately and very slowly is how lichens build themselves across stone. A lichen is not a single organism. It is a partnership of fungus that has learned to cultivate algae or cyanobacteria within its own tissue, housing the photosynthetic partner, in exchange for a share of the sugars that partner produces from light. The fungus provides structure, moisture retention, and mineral access. The algae provide energy. Together, they form something neither could build alone. An organism capable of colonizing bare rock in environments where nothing else survives. Arctic tundra, Antarctic dry valleys, the surfaces of buildings in cities, the vertical faces of cliffs with no soil and no shelter. In some of these environments, lichens are the only visible life patches of gray and orange and pale green on surfaces that appear, to a passing eye, to be simply stone. Their growth rate is so slow that a crust the size of your palm may have been building for a century. Researchers use this slowness deliberately, dating the age of rock surfaces exposed by glacial retreat by measuring the diameter of lichen colonies on them. The lichen becomes a clock, one calibrated not in seconds or years, but in the patient accumulation of fractions of millimeters, recording the passage of time in a language written on stone. Stone holds other records. In pieces of amber fossilized tree resin from forests that no longer exist, researchers have found mushrooms preserved in extraordinary detail. The amber from the Cretaceous period, nearly 100 million years old, contains fungal specimens with their caps and gills and spore surfaces intact, frozen at the moment when the resin flowed over them and enclosed them permanently. What those specimens show is a form that is, in its essential architecture, recognizable. The mushroom shape, the dome, the stem, the radial arrangement of gills beneath, was already established 100 million years ago and has not changed in any fundamental way since. The forests those mushrooms grew in have been replaced, the trees transformed through millions of generations of evolution, the insects and animals that walked among them replaced by entirely different lineages. The mushrooms kept their form, whatever pressures reshaped everything around them, across 100 million years of mass extinctions and continental drift and climate upheaval. The basic fungal architecture found its solution early and stayed there. A mushroom in Cretaceous amber looks like a mushroom you could find in a forest tonight. That continuity, across that span of time, is one of the quieter forms of permanence the natural world contains. Contained within the spore cases of bird's nest fungi is a mechanism so precise it seems to belong more to physics than to biology. These small fungi, rarely taller than a fingernail, shaped like tiny cups or goblets, hold their spores in enclosed packets called peridioles, which sit inside the cup like eggs in a nest. When a raindrop falls into the cup at the right angle with sufficient force, it does not simply splash. The geometry of the cup converts the drop's energy into a directed ejection. The peridiole is launched outward and upward on a thin cord of fungal tissue that unspools as it travels, until the cord catches on a nearby twig or stem and the peridiole swings around it, wrapping tight. The spore packet is now attached to vegetation at a height where grazing animals are likely to consume it, and the spores pass through the animal's digestive system unharmed, to be deposited elsewhere with the dung. This sequence, cup geometry, raindrop physics, ballistic ejection, cord mechanics, animal dispersal, is not a series of adaptations that developed separately and happened to work together. It is a single integrated system, each part dependent on the others, refined over millions of years into something that functions as reliably as a machine. A raindrop falls, a spore travels. The mechanism has no moving parts except water and the physics of impact. Impact on human history has come from fungi more than once, and not always gently. Ergo is a fungus that infects grain rye, in particular, replacing the seed with a dense mass of fungal tissue that contains, among other compounds, alkaloids related to the ones later used to synthesize lysergic acid diethylamide. When ergot-infected grain was milled into flour and baked into bread, the compounds survived the heat and entered the bodies of everyone who ate from the loaf. Mass poisonings occurred across medieval Europe, whole villages experiencing convulsions, hallucinations, a burning sensation in the limbs. The condition was called St. Anthony's fire. Historians have proposed ergot poisoning as a contributing factor in episodes of collective hysteria, including events in Salem in the 1690s where accusations of witchcraft followed a winter of contaminated rye. The connection is debated and not conclusively established. What is established is that a fungus, growing invisibly on a staple crop, introduced chemistry into the human nervous system that altered behavior at a community scale, and that the people experiencing these effects had no framework for understanding what was happening to them except the frameworks their culture provided. A fungus shaped the course of those events, quietly, without intention, as fungi always do. Always doing their work without acknowledgement is something fungi have practiced for longer than almost anything else alive. The fossil record extends their presence back to a period so early that the continents were arranged in shapes no modern map would recognize. Fossilized fungal filaments have been found in rock formations, approaching 1 billion years in age, a span of time that precedes complex animal life, precedes the colonization of land, precedes almost every organism whose name a person might recognize. These were not primitive organisms in the sense of being simple or incomplete. They were fungi, fully fungal, threading through ancient ocean sediments, with the same basic chemistry and the same basic strategies their descendants use today. 1 billion years of refinement has not changed the fundamental approach. It has only deepened the range of environments those fundamentals can access. Every habitat fungi now occupy every forest floor, every rotting log, every nuclear ruin, every deep ocean trench. Every Antarctic rock was reached by the same essential organism that was already threading through primordial sediment before the first animal drew its first breath. The kingdom of fungi did not begin with the world as we know it. It was present, already patient, already working, while the world we know was still assembling itself. There is a mushroom that smells like warm maple syrup, not approximately, not distantly precisely, like the syrup that pools at the edge of a stack of pancakes on a cold morning. The candycap mushroom, found in coastal forests of the American West, produces lactones, volatile compounds that the human nose reads as sweetness, as warmth, as something domestic and familiar. Chefs use dried candy caps in ice cream and shortbread, in sauces and custards, transforming what grew from the base of a tan oak into something indistinguishable from a confection made with cane sugar and maple trees. The mushroom did not evolve this scent for kitchens. It evolved it, most likely, as a way of attracting the insects and small animals that would carry its spores outward. The fact that the scent it arrived at overlaps so precisely with one of the most comforting flavors in human cuisine is coincidence in the technical sense. But it is the kind of coincidence that makes the boundary between the forest and the familiar feel unexpectedly thin. To stand in a coastal woodland in autumn and catch, without warning, the smell of breakfast, that small, disorienting comfort, arriving from exactly the wrong direction, is to be reminded that the chemistry of pleasure does not belong exclusively to kitchens. It was in the forest first. First, to feed on what most organisms cannot touch. Certain fungi approach wood from the inside. Mycelium enters a fallen log, not through its surface but through cracks, through the tunnels left by insects, through any discontinuity in the bark that offers a point of entry. Once inside, it begins dismantling the wood structure from within, secreting enzymes that break the lignin and cellulose into simpler compounds, absorbing the released sugars pushing threads further into the heartwood. From the outside, the log appears intact for months or years. The bark holds, the shape holds, but inside, the transformation is already well advanced, the dense wood becoming lighter, softer, its interior threading with pale mycelium, until what looked like solid timber is revealed, when you lift a corner of bark, to be something approaching a network in its own right. This is not destruction. It is conversion. The carbon and nitrogen and phosphorus locked inside that tree are being returned to the soil in forms that roots can absorb, not wasted, not lost, but moved from one part of the cycle into the next. The log disappears over years, and what it becomes is the ground around it, slightly enriched, slightly darker, slightly more capable of supporting the seedling that has already taken root in the shadow the log casts. Casting light instead of shadow is what bioluminescent fungi do in the hours after dark. A patch of decaying wood on the forest floor that appears unremarkable in daylight grey, soft, partially buried in leaf litter, may, in total darkness, emit a faint and steady greenish glow from the mycelium threading through it. The light is cold, produced without heat by the oxidation of a compound called luciferin. In the presence of an enzyme called luciferase, it costs the fungus very little energy. It continues for as long as the chemical reaction has substrate hours, days, weeks. Some species glow most intensely at the moment of peak spore release, which has led researchers to propose that the light attracts nocturnal insects, who then carry spores to new locations. Other species glow from underground mycelium where no insect would see them, which leaves the function uncertain. Scientists name this uncertainty honestly. Whatever the function, the phenomenon is real, verifiable, photographable, present in forests across multiple continents. To walk into a dark forest and find the ground emitting its own quiet light, without source, without flicker, without the logic of fire or electricity that experience sits outside the categories most people carry for what the natural world does at night. The dark, it turns out, is not always dark. Dark in a different sense is the interior of a mycelial network processing what it has encountered. Researchers studying how fungal networks respond to spatial problems have grown mycelium across surfaces, representing geographic regions, nutrients placed at points corresponding to population centers, barriers representing mountains or water. In each experiment, the network produces, without any external guidance, a system of connections that closely resembles the infrastructure humans designed for those regions after decades of engineering analysis. When a path is blocked, the network reroutes. When two rich sources exist, it builds redundancy. The shapes are not approximate matches. They are measurably similar to human-designed systems in their efficiency metrics, the ratio of path length to direct distance, the distribution of node connectivity, the placement of backup routes. No single part of the mycelium knows the whole map. Each thread follows only local chemical gradients, grow toward food, avoid toxins, fuse with compatible threads nearby. From these simple rules, operating simultaneously across the whole network, efficient global structure emerges. Engineers studying this are not doing so out of curiosity alone. Algorithms derived from observed mycelial behavior are being tested as design tools for road networks, communication infrastructure, and supply chains. The forest has been solving these problems continuously for hundreds of millions of years. We are only recently paying close enough attention to learn from the solutions it has already found. Already found in the human gut, though we rarely think of it this way, is a community that includes fungi alongside bacteria. The microbiome, that vast population of microorganisms living within the digestive system, contains fungal species that interact with the bacterial community, with the immune system, and through mechanisms still being mapped, with the nervous system that connects the gut to the brain. Researchers studying the gut-brain axis have found that the composition of the microbial community, including its fungal members, correlates with mood, with anxiety levels, with the body's response to stress. The direction of causation is not yet fully established, whether fungal imbalance contributes to psychological states, or whether psychological states alter the fungal community, or whether both are downstream of other factors. Scientists hold these findings carefully, without overclaiming. What is clear is that fungi are present inside you, right now, as you rest, not as invaders, not as pathogens in this context, but as members of a community your body has maintained for your entire life. You have never not had fungi inside you. They are part of the ecology of a human body, as they are part of the ecology of a forest, present, functional, woven into a system too complex to fully see from any single vantage point. From a single vantage point on a forest path, the mushrooms you can see are a small and unrepresentative sample of the fungal activity occurring within range of your vision. Most of what fungi do happens out of sight, inside wood, underground, within root tissue, in the spaces between soil grains. What surfaces as a mushroom is the fruiting body, the reproductive structure, the visible moment of a process that is mostly invisible. And the fruiting body, once it has released its spores, dismantles itself. Some species do this through a process called autodigestion. The cap and gills dissolve from the edges inward, the tissue liquefying through the action of the fungus's own enzymes, the black fluid dripping from the margin of the cap as the structure returns its materials to the environment. An inky cap mushroom, found one morning standing white and fresh in damp grass, may by evening have dissolved to a puddle of dark ink at the base of its stem. The process takes hours. It is complete, tidy, and leaves nothing behind that does not belong to the soil. The mushroom does not persist beyond its function. It completes what it came above ground to do, and then it undoes itself, returning everything it borrowed from the earth with a thoroughness that no human structure achieves. Achieving thoroughness of a different kind, certain fungi have developed relationships with specific tree species, so exclusive they cannot be transferred. The Matsutake mushroom forms mycorrhizal partnerships with particular pines, and only those pines in a specificity so narrow that it cannot be cultivated commercially, despite being among the most valuable edible fungi in the mycelium world. Where the trees grow, the mushroom may grow. Where the trees are absent, it will not. The relationship took millions of years to develop and is encoded so deeply into the chemistry of both organisms that it cannot be reconstructed under artificial conditions. Researchers have tried. They have grown the trees, prepared the soil, introduced the spores under controlled conditions that replicate every measurable variable of the natural environment. The mushroom does not fruit. Something essential to the partnership remains unmeasured or unmeasurable or simply beyond current replication. This exclusivity is not unusual in the fungal kingdom. Many mycorrhizal species show strong preferences for particular hosts. Their networks intertwined with specific root chemistries in arrangements that evolve together over timescales no experiment can compress. The fungus and the tree are, in a sense that goes beyond metaphor, made for each other, not by intention, by the slow accumulation of co-evolutionary time. Time accumulating in a different register is visible in the rings of a fairy ring's slow expansion, and in the geometry of how mushrooms arrange themselves when they fruit in groups. Arcs, spirals, irregular curves that follow the advancing edge of the underground mycelium, the shapes that emerge, are governed by the mathematics of even expansion, the same mathematics that produces the spiral of a nautilus shell and the arrangement of seeds in a sunflower head. When the expansion is unobstructed and the soil is uniform, the result is a circle. When the expansion meets a barrier, a stone, a tree root, a change in soil composition, the mycelium bends around it and continues, producing an arc or an interrupted curve that maps the underground obstacles as precisely as a survey. The mushrooms at the surface are, in this sense, a readout of the underground terrain, a map drawn not by intention, but by the physics of growth against resistance. To see a curved line of mushrooms crossing a lawn is to see a record of what the soil contains below the surface, at the depth where the mycelium travels. The pattern above is a translation of the pattern below. The mushroom is reading the ground for you, in a language made of caps and stems and the spaces between them. Between one species and the next, fungal chemistry varies in ways that no other kingdom of organisms matches for range and surprise. Some mushrooms produce compounds currently being studied as treatments for depression, psilocybin, a molecule that the human nervous system converts into a form that binds to serotonin receptors with an affinity and duration that produces, in clinical settings, effects that standard antidepressants do not replicate. Early trials in people with treatment-resistant depression have shown results that are, by the careful language of clinical research, promising. The studies are small, the mechanisms are incompletely understood. The regulatory frameworks for therapeutic use are still being developed. Researchers hold all of this with appropriate caution, naming what is known and what remains open. What is already established is that a molecule produced by a mushroom growing from forest soil interacts with the human brain in ways that may alleviate suffering that other treatments cannot reach. The mushroom evolved psilocybin. Most likely, as a defense against insects, the compound disrupts the nervous systems of invertebrates. It did not evolve it for human therapy. And yet here we are, studying what the forest made for its own purposes, and finding in it something that may serve ours. Serving a purpose the forest did not plan for them is what fungi do every time they are brought into human spaces. Yeast, single-celled fungi, invisible, weightless, drifting in the air of every kitchen and bakery, were discovered not through deliberate search, but through the simple fact of their presence. Crushed fruit left in a warm place ferments. Wet grain left overnight develops bubbles. Dough mixed and rested rises. The organisms responsible for these transformations were present in the environment long before humans understood what they were, and the techniques that use them developed through observation of outcomes rather than understanding of mechanisms. Every sourdough starter, that small pot of fermenting flour and water that bakers maintain and feed and adjust over years, developing its specific character, is a community of wild yeast and bacteria that assembled itself from whatever was present in the flour, the water, the baker's hands, the air of that particular kitchen in that particular place. No two are identical. The yeast that makes bread rise in one city is not quite the same population as the yeast doing the same work in another. The fermentation that produces wine in one valley differs subtly from the fermentation in the valley adjacent. Fungi have been shaping human culture since the first deliberate fermentation, without any awareness that culture existed. They were simply doing what they do, consuming sugars, releasing gas, continuing the chemistry of their own survival, and everything else followed. On the underside of a mushroom cap, if you look closely enough to see the structure rather than simply the surface, the gills are arranged with a precision that took researchers by surprise when they began measuring it. The spacing between adjacent gills consistent, regular, narrowing toward the stem and widening toward the cap edge in a gradient that maximizes surface area, while maintaining airflow between them, has been studied as a model for ventilation engineering. The geometry allows air to move between the gills by convection, drawing spores downward and outward without requiring wind. Change the spacing slightly in either direction, and the efficiency drops. The mushroom arrived at this geometry through evolution rather than calculation, but the result is optimal, in the same sense that engineered solutions are optimal. It achieves maximum function with minimum material. Architects studying natural ventilation systems have found the gill geometry useful as a reference for designing spaces that move air efficiently without mechanical assistance. The underside of a mushroom cap, examined not as texture but as structure, is a ventilation system refined over millions of years of selection pressure into something that functions, by measurable metrics, about as well as it is possible to function. The forest floor is scattered with them. Millions of small, precise, quietly optimal structures, opening and closing with the seasons, doing their work without acknowledgement. Acknowledgement came centuries late for the role fungi play in producing the bread that has sustained human populations across most of recorded history. Every culture that developed grain agriculture eventually developed fermentation alongside it, and every fermentation relied on fungi. The sourdough traditions of Egypt, the leavened breads of ancient Rome, the fermented grain drinks of pre-Columbian Mesoamerica, all of them depended on yeast. And yeast is a fungus, and the fungus was never invited. It arrived on its own, drawn by the sugar of crushed grain, establishing itself in the warm, wet environment of early food preparation, and the humans who observed the results learned to maintain the conditions that produced them. This is not a peripheral contribution to human civilization. Leavened bread, bread with the texture and digestibility that fermentation produces, made grain more calorie accessible, more palatable, more storable. It contributed to the dietary foundation that supported the population growth and energy surplus that allowed cities, writing, and organized culture to emerge. The connection is not direct or simple, but it is real. Fungi did not build civilization, but the chemistry they perform on sugar was present at the foundation of the conditions that made it possible. The yeast did not know. It was simply hungry, as it always is, and the results were larger than anything that required intention to produce. Producing results larger than their size suggests is something fungi accomplish through chemistry that remains, in certain cases, genuinely beyond human synthesis. Some fungal compounds molecules produced through metabolic pathways that evolved over hundreds of millions of years have not been replicated in any laboratory. Researchers can identify their structure, can trace their effects in biological systems, can study how they interact with receptors and enzymes and cell membranes. But the synthesis, the step-by-step chemical construction of the molecule from simpler components, remains beyond current capability for a small but significant number of fungal products. This is not for lack of trying. Organic chemistry has made extraordinary progress across the 20th and 21st centuries, and most complex natural molecules have eventually yielded to synthetic approaches. But some fungal compounds involve chiral arrangements, ring structures, and functional groups assembled through enzymatic processes that no current synthetic route can efficiently reproduce. The forest is producing chemistry that our best laboratories cannot yet make. Some of what fungi know how to build, we do not yet know how to ask them to teach us. Teaching through observation rather than instruction is how human knowledge of medicinal fungi accumulated across thousands of years. In traditional Chinese medicine, reishi, a hard, lacquered bracket fungus that grows from the base of old trees, has been called the mushroom of immortality for over 2,000 years. The name is not literally meant. What it points toward is a consistent observation made by practitioners across centuries that preparations from reishi appeared to support the immune system, reduce inflammation, and improve the quality of sleep and rest in people who used them regularly. Modern pharmacology has found compounds in reishi triterpenes, beta-glucans that interact with immune signaling in ways consistent with those traditional observations. The mechanism is not yet fully understood. The clinical evidence in humans is promising but not yet definitive by the standards of controlled trials. Researchers hold this gap honestly, noting what traditional use suggests and what laboratory evidence supports, without collapsing the distance between them. What sits in that gap between centuries of careful empirical observation and the beginnings of mechanistic explanation is a reminder that knowledge about the natural world accumulated through sustained attention long before it accumulated through controlled experiment. The forest was a laboratory before laboratories existed. Existing at the boundary between living and built is what mycelium does when it is grown into architectural forms. Researchers and designers have developed processes for growing fungal tissue through agricultural waste hemp herds, corn stalks, sawdust, into molds that produce finished objects when the growth is complete and the material is heat-dried. The resulting substance is rigid, lightweight, and fully compostable. Its mechanical properties, compressive strength, impact resistance, thermal insulation can be adjusted by varying the species of fungus used, the substrate composition, and the growth conditions. Buildings insulated with mycelium panels are already standing. Packaging grown from mycelium has already shipped products across continents. Furniture assembled from mycelium components already exists in homes. What makes this material different from other bio-based alternatives is that it does not require harvesting. It grows into its final shape, with no cutting, no waste, no manufacturing residue. The fungus does the forming. The designer provides the mold and the conditions. Everything that happens between those inputs and the finished object is biology, operating according to its own logic in the dark. The built environment is beginning, slowly, to include structures that were grown rather than made, and the organism doing the growing is one that has been building structures in forests for hundreds of millions of years. For hundreds of millions of years, certain partnerships have operated below the threshold of what either partner could achieve independently. Mycorrhizal fungi connected to the roots of trees in a healthy forest do more than exchange nutrients. They provide a form of biological immunity. When a pathogen attacks the roots of a connected tree, the fungal network can facilitate the movement of defensive compounds from unaffected neighbors, compounds that prime the attacked tree's own immune response before the pathogen has fully established itself. Trees with intact fungal connections survive root pathogen attacks at higher rates than isolated trees. The network is not a medical system in any designed sense. It is an ecological consequence of connection. The same threads that move phosphorus can move signal molecules, and signal molecules can carry information about threats. The forest does not plan its own defense. It simply maintains its connections, and from those connections, a kind of distributed resilience emerges. Remove the fungal layer, and the individual trees become more vulnerable, each standing alone against whatever arrives. Maintain it, and the forest behaves, in moments of stress, more like a single organism with distributed immune function than like a population of separate individuals, each managing its own survival. Managing survival in the most extreme conditions the planet offers is something certain fungi have turned into their defining characteristic. In the dry valleys of Antarctica, where liquid water is present for only a few weeks of the year, and temperatures fall below the point where most biological processes can continue, fungi persist. Inside rocks, within the thin films of moisture that form beneath translucent quartz, in the frozen soil that thaws for days, rather than months they are there, metabolizing slowly, growing fractions of a millimeter per year, maintaining the minimum of biological activity needed to remain alive. What allows this is partly the flexibility of fungal metabolism, the ability to slow cellular processes to nearly undetectable levels without ceasing them, and partly specific adaptations like the production of compounds that prevent ice crystals from forming inside cells. These antifreeze molecules are not unique to Antarctic fungi, but they are present in higher concentrations in the species that occupy the coldest habitats. The fungus does not fight the cold. It accommodates it, adjusting its internal chemistry to work within conditions that would freeze and rupture the cells of most organisms. Cold, for these fungi, is not the enemy. It is simply the parameter within which survival must be arranged. Arranged with patience is also how certain fungi approach the question of when to fruit. Some species remain entirely underground or inside wood, or within root tissue for years between visible appearances. The mycelium persists, grows slowly, extends its network, accumulates resources. Then, when some combination of temperature and moisture and soil chemistry aligns in the configuration the species requires, fruiting bodies emerge. A log that appeared bare for 4 years suddenly produces dozens of mushrooms across its surface in a single week. A patch of forest floor that has shown nothing for a decade erupts briefly with caps after a particular rain following a particular dry summer. The triggers are species-specific and not always fully understood. What is understood is that the waiting is not dormancy. In the way a seed is dormant, the mycelium is active throughout, maintaining itself, responding to its environment, simply not fruiting. The decision to fruit, when it comes, is a response to a specific configuration of conditions that the network has been monitoring continuously. The mushroom does not appear because conditions improved. It appears because conditions became precisely right, a distinction that took the mycelium years of quiet assessment to determine. Determining the structure of a mycelial network from the outside is nearly impossible without specialized imaging. From above, a forest floor shows leaf litter, soil, the occasional surface root. Nothing reveals the extent of what lies beneath the threads extending through meters of soil in every direction, connecting root systems that appear, from the surface, to be entirely separate. Researchers mapping these networks using DNA analysis of soil samples have found that a single fungal individual can extend across areas larger than a football field, its threads present in soil samples taken from dozens of points across the site. The organism has no center. There is no location where it is most itself, no core from which the threads radiate. It is distributed across its entire extent with equal presence, as much itself at the periphery as at any other point. This is a form of existence that has no clear human equivalent. We experience ourselves as located here, in this body, at this point in space. The mycelium experiences, if experience is even the right word, something closer to being everywhere it is simultaneously, with no hierarchy of location, no sense of center and edge. It simply occupies the space it occupies, fully, at every point. Every point in a forest is connected to every other point through threads too fine to see, and old enough to have been present before the oldest trees were seedlings. Some mycelial networks are older than the forests above them. The fungus arrived first in soil that was different then, thinner, less developed, supporting different vegetation, and the forest grew up around and within an existing fungal architecture. The trees that seem to anchor the forest, their roots going deep, their trunks defining the space they are, in a certain light, inhabitants of a fungal structure that predates them. When a tree falls and its roots decay, the mycelium that was partnered with those roots does not fall with it. It persists, redirects, partners with whatever grows in the gap. When a fire moves through and the canopy is lost, the mycelium remains in the soil below the burn, waiting for the first seedlings to emerge into the cleared light so that the partnerships can begin again. The forest you see, the one defined by its trees, by its canopy, by the way light falls through the branches in the morning, is the visible layer of something older and less visible and more continuous. The trees come and go. The fungal network beneath them, in the forests where it has been left undisturbed, simply continues. Pull back the bark of a fallen log in a temperate forest, carefully, slowly, the way you might lift the cover of something that has been resting undisturbed, and what you find beneath is not emptiness. The wood is threaded with white, pale filaments running along the grain of the timber, following the channels that water and nutrients once traveled through the living tree, now repurposed as corridors for the mycelium dismantling the wood from within. The lignin that made the wood hard, the molecule that gave the tree its structural rigidity, the compound that no other kingdom of organisms can fully break down, is being taken apart here, enzyme by enzyme, in a process so chemically sophisticated that researchers studying it have found applications in paper manufacturing, biofuel production, and the development of new textile fibers. The fungus does not know it is performing chemistry of industrial relevance. It is simply feeding, and in feeding, it is doing what it has done since the Carboniferous period, When forests first evolved wood and fungi, eventually evolved the means to return that wood to soil. Every fallen log is a slow laboratory, conducting experiments in molecular disassembly that began before humans existed and will continue long after any particular human concern has passed. The white threads are not a sign of loss. They are the sign of conversion matter moving from one form into the next, as it always has, as it always will. Matter moving through a system whose true scale remains difficult to hold in the imagination. Beneath a stand of conifers in the Pacific Northwest, the soil may contain more living fungal tissue by weight than all the above-ground vegetation combined. The trees, some of them decades old, their trunks wide enough to shelter under, appear to be the substance of the place, the dominant presence, the living weight of the forest. But measure the mycelium threading through the soil below them, and the numbers shift. The forest's center of biological gravity is underground. What grows above is the visible portion of a system whose greatest mass and activity is hidden, operating in the dark, in a medium that most people never think to consider as habitat. A handful of healthy forest soil contains more fungal thread than would stretch the length of several city blocks if laid end to end. The soil is not the ground beneath the forest. In a sense that is more than poetic, the soil is the forest's body, and the fungal threads are its tissue, and the trees rising above are the visible expression of something whose main existence is below, in the dark, in the quiet, doing work that the canopy above depends on entirely, quietly doing work that heat alone completes. This is what certain fungi require before they can be received by the body. The moral mushroom, honeycomb-capped and hollow-stemmed, one of the most prized edible fungi in the world, is mildly toxic when raw. The compound responsible breaks down under heat. A morel eaten directly from the forest floor causes nausea. The same morel sautéed for 4 minutes in a warm pan becomes something chefs describe as among the most intensely flavored ingredients available: nutty, earthy, with a depth that persists in the mouth long after the bite. The transformation is chemical. Heat dismantles the toxic compound into harmless components, while simultaneously triggering reactions across the morel's complex surface geometry, producing flavor compounds that could not form any other way. What is dangerous raw becomes extraordinary cooked. Several other fungal species share this property chemistry that responds to heat in ways that make the result worth the knowledge of how to apply it. That knowledge was discovered and refined and passed down across generations of people who ate from the forest and learned, through careful attention over long time, what the forest required of them before it offered what it had. What it had to offer, the human nervous system arrived in ways no one anticipated. Psilocybin, present in a group of mushrooms distributed across multiple continents, converts in the body into a compound that binds to serotonin receptors with an affinity that produces effects no synthetic molecule fully replicates. Clinical trials for treatment-resistant depression, for end-of-life anxiety, for addiction, these are ongoing, their results described cautiously by researchers not given to overstatement as significant. Participants frequently report something that existing pharmacological language struggles to capture: a sense of connection to other people, to the natural world, to something larger than the experience of being a single person moving through a particular life. Researchers note this without yet explaining it mechanistically. The molecule appears to temporarily alter how the brain maintains the boundary between self and world loosening. Briefly, something that is usually held tightly. The mushroom did not evolve psilocybin to offer this. It evolved it, most likely, as a defense against insects, whose nervous systems it disrupts at much lower doses. The human experience of it is incidental to its origin, and yet the experience is real, and the molecule is ancient, and the forest has been producing it continuously for longer than our species has existed to encounter it. Encountering the forest at night changes what is visible. Certain mushrooms glow most intensely at the precise moment they are releasing spores, peak luminescence aligned with peak reproductive activity, the light attracting nocturnal insects that depart carrying spores on their bodies. The mushroom has built a signaling system, operating in the one part of the day when a faint green light on the ground is the most visible thing within range of a flying beetle. Whether all bioluminescent species use their light this way is genuinely uncertain. Some glow only from their mycelium, deep inside rotting wood, where no insect would find it. The function there remains unknown. Researchers hold this honestly, noting where the evidence runs out. What does not run out is the phenomenon itself, verifiable, photographable, present in forests across multiple continents. To walk into a dark forest and find a patch of ground emitting its own quiet light, without fire, without electricity, without any of the sources the mind reaches. For first, that experience sits outside the categories most people carry for what the natural world does after dark. The forest lights itself at night in ways that existed long before anyone was there to notice them, and will continue long after the last observer has gone home. Gone from the meadow by morning is what certain fungi accomplish in hours rather than years. The inky cap mushroom found in damp grass, white and fresh in the early light, dissolves itself. Not through external decay, but through autodigestion. The cap and gills liquefying from the edges inward, through the action of the fungus's own enzymes, the tissue darkening and dripping as the structure returns its materials to the environment, in a process complete within a single day. A mushroom found standing at dawn may be a puddle of dark ink by evening, leaving nothing behind that does not belong to the soil. There is a tidiness to this that no human structure achieves. The mushroom does not persist beyond its function. It completes what it came above ground to do, releasing spores during the hours when it stands, and then systematically undoes itself, returning everything it borrowed from the earth without remainder. This is not failure. It is the closing of a loop. The mushroom's entire above-ground existence is organized around one task, and when the task is done, the structure that performed it quietly ceases, contributing its materials to whatever comes next in the same patch of soil, in the same continuous cycle that has been running since before the meadow existed. Running beside that cycle, invisible and continuous, is what the mycelium does when the forest is stressed. Chemical signals propagate through the fungal threads when part of the network is damaged or threatened, a section severed by a burrowing animal, a root under pathogen attack, a tree losing ground to drought. The change ripples outward through the connected threads, altering their chemistry in ways that reach organisms meters away. Those organisms adjust. Resources shift. Defensive compounds move toward the threat. The network does not plan this response or coordinate it through any central point. It emerges from the chemistry of individual threads reacting to local conditions, those local reactions accumulating across the whole into something that looks, from outside, like a unified reply. Ecologists have found that forests with intact mycelial networks recover from disturbance faster than forests where those networks have been fragmented. The connection is part of the defense. What the threads carry is not only nutrients, but the information that allows a community of separate organisms to behave in moments of pressure, as though they share a nervous system. They do not share a nervous system. They share something older and less understood, a chemical conversation conducted through living threads in the dark, continuous, below the surface, older than any forest currently standing above it. Above it, the fly, agaric, announces itself in the most legible visual language the fungal kingdom possesses. Red cap, white spots, the archetypal mushroom shape, this is Amanita muscaria, the image that appears in fairy tale illustrations, on holiday decorations, in the oldest shamanic traditions of Siberia, where practitioners consumed preparations of it to enter states understood as contact with non-human realms. The mushroom contains ibotenic acid and muscimol, compounds that affect the nervous system in ways distinct from psilocybin, producing experiences interpreted through the frameworks of the cultures that used them. How the red-capped mushroom entered European folklore, whether through the westward spread of shamanic symbolism, through independent discovery, through mechanisms not yet traced, is still debated. What is not debated is the reach of the image, recognized across a geographic range too wide to explain by coincidence, embedded in visual traditions that developed without contact with one another. The mushroom found its way into human symbolic life in multiple places independently. Or, the human nervous system, encountering this particular fungus across its range, responded in ways that left similar marks across different cultures. Either way, the image persists on greeting cards, in children's books, in the oldest recorded ceremonies, still carrying something that rational explanation has not fully discharged. Discharged into the atmosphere in quantities that affect how rain forms is what fungal spores do when they rise on warm air above a forest canopy. Spores released from the ground drift upward in thermals, reaching altitudes where they encounter the conditions that allow water vapor to condense around them. A spore surface provides the nucleation point, the physical anchor around which a water droplet can begin to form. Forests release spores in vast quantities throughout the growing season, and those spores entering the upper atmosphere may seed cloud formation over the same forests that produced them, drawing rainfall back down onto the ground that released the spores in the first place. The system is circular in a way that researchers are still carefully quantifying. The evidence that spores contribute to cloud nucleation is established. The degree to which this contribution shapes regional precipitation patterns is under active investigation. What the research so far suggests is that a healthy forest may participate in maintaining its own water supply, that the spores it releases into the air return as the rain it needs to fruit and grow and release more spores. The forest does not intend this cycle. It simply performs it continuously, and the water falls where the spores were. Where the spores were is sometimes a place no mushroom was expected. Deep ocean trenches, lightless, cold, under pressures that would collapse any unprotected human body, have yielded fungal samples to researchers who deployed collection equipment at depths of several thousand meters. These are not surface fungi that drifted down passively. DNA analysis reveals species adapted to deep marine conditions, their metabolisms oriented toward the chemical energy available at those depths, rather than the photosynthetically derived energy that powers most of what we think of as life. Their relationship to the broader fungal kingdom, whether they share recent common ancestry with terrestrial species or represent ancient lineages that never left the ocean, is still being worked out. What they demonstrate is that the boundary conditions for fungal life extend further than the most generous previous estimates. Cold, dark, pressurized, chemically alien conditions that define the deep ocean floor are conditions under which fungi not only survive but apparently thrive, doing whatever it is fungi do in the dark at the bottom of the sea, in a silence so complete and a pressure so absolute that the rest of the living world is entirely absent. They are there, quietly, as fungi always are, doing their work in a place no one thought to look until recently enough that the surprise is still fresh. A mycelium composite grown in a mold and then heat-dried produces a material that engineers describe, when they first handle it, as unexpectedly substantial. Not heavy lighter than most plastics, but dense in a way that suggests structural integrity rather than fragility. Companies manufacturing packaging from it report that it absorbs impact better than expanded polystyrene, insulates more effectively than many petroleum-based foams, and returns to soil within weeks when composted. The fungus that produced it was given agricultural waste, corn husks, hemp fiber, sawdust as its substrate, and it threaded through that waste, binding it, filling its gaps, knitting the loose material into a unified structure through the same process it uses to colonize a fallen log. No adhesive was added, no synthetic binder. The binding is biological, produced by the mycelium as a natural consequence of its growth. When the growth was complete and the material was dried, what remained was something that took millions of years of evolution to develop and approximately 2 weeks to grow. Architects are exploring it as insulation. Furniture designers are growing chairs and table surfaces from it. The built environment is beginning, slowly and experimentally, to include structures that were cultivated, rather than manufactured, grown in the dark, by an organism that has been building structures in forests since before the first tree existed. On the surface of a rock face in the high Arctic granite, pale gray, angled slightly south, to catch whatever light the season offers, a lichen, the diameter of a dinner plate, may have been growing since before the printing press was invented. Its edge advances a fraction of a millimeter per year or less. In winter, it freezes solid and waits. In the brief Arctic summer, it photosynthesizes for a few weeks when moisture permits. Then it freezes again. This is its year, repeated without variation for centuries. Researchers use this slowness deliberately. By measuring the diameter of lichen colonies on rock surfaces exposed by glacial retreat, they can estimate when the ice last covered that ground. The lichen becomes a clock, one calibrated not in seconds or minutes, but in the patient accumulation of almost imperceptible growth, recording the passage of geological time in a language written directly on stone. A gravestone in a churchyard covered in lichen is not being decorated, it is being measured. The lichen arrived after the stone was carved and will remain after the inscription has worn to illegibility, still advancing its edge by fractions of a millimeter each year, indifferent to what the stone once said, attending only to the chemistry of the mineral surface and the angle of available light. Some fungi seal the wounds of living trees. When bark is damaged by a falling branch, by an insect boring through to the cambium, by any of the small injuries, a tree accumulates across a long life. Certain fungal species enter through the breach and produce resins and compounds that harden within the damaged tissue. The tree's own response to injury is augmented by the fungus, the wound site becoming a collaborative structure, partly tree, partly fungal, that resists further pathogen entry more effectively than either partner's defenses alone. This is not a relationship that developed recently. The partnership between wound-sealing fungi and the trees they protect has been operating for long enough that the chemistry of both organisms reflects it. The tree's wound response includes signals that attract compatible fungal partners, and the fungi produce compounds specifically suited to the biochemical environment of tree damage. The forest does not experience injury the way an isolated organism does. It experiences injury within a network of relationships that begins responding before any individual organism has fully registered what happened. A damaged tree in a connected forest is not simply a damaged tree. It is a damaged node in a system that has already begun the process of repair quietly, underground, through threads that were present before the damage occurred. In a laboratory in Japan in the early 2000s, a researcher named Toshiyuki Nakagaki placed a slime mold, a fungus-like organism, at one entrance of a maze and food at another. The organism solved the maze. Not quickly, not elegantly at first, but within hours it had retracted its growth from dead ends and concentrated its tissue along the shortest viable path between start and food. The experiment was repeated with mycelium of true fungi, with similar results. Then researchers mapped the Tokyo rail network onto a flat surface, placed nutrients at points corresponding to population centers, and watched what the mycelium produced. The network it built matched the actual rail system, with a fidelity that engineers found uncomfortable in its implications. The fungus was not planning. It was following chemical gradients, each thread responding only to local conditions, the global solution emerging from the accumulation of local decisions made simultaneously across the entire network. This is precisely how certain optimization algorithms now used in network design were developed not by studying fungi, but arriving independently at the same mathematical logic. The forest had the answer before the question was formally asked. The mycelium was solving infrastructure problems in the soil of ancient forests, while the cities those systems now serve were still centuries from being built. The human gut has never been without fungi. From the first days of life through birth, through early contact with skin and air and food, fungal species establish themselves in the digestive system alongside the bacterial community, and they remain there, adjusting in composition as diet and environment shift throughout the entire span of a human life. This fungal component of the microbiome is called the mycobiome, and its study is considerably younger than the study of gut bacteria. Researchers are finding that the fungal community interacts with the bacterial one in ways that affect both, competing with some species, supporting others, producing compounds that influence the immune cells lining the gut wall. The gut immune system, which must constantly distinguish between harmful pathogens and the beneficial residents it should leave undisturbed, appears to calibrate partly against the fungal community it encounters in early life. Disruptions to the mycobiome, through antifungal medications, through dietary changes that alter the substrate available to different species, have been correlated with shifts in immune function and inflammatory markers. The relationship is complex, and the research is early. What is not early is the relationship itself. Fungi have been part of the human gut community for as long as humans have existed, and for longer than that. The gut microbiome of our primate relatives suggests the partnership predates our species by millions of years. A single hypha-1 thread of mycelium, the basic unit from which all fungal networks are built, is so fine, it passes through the spaces between individual soil grains without displacing them. It navigates the microscopic architecture of the ground, the way water finds the path of least resistance, not by force, but by following the geometry of what is already there. And yet, from these individual threads, gathered in their billions across a forest floor, something vast and responsive emerges. The transition from thread to network is one of the stranger phenomena in biology, each hypha operating according to simple local rules. No single thread aware of the whole, and yet the whole exhibiting behaviors that no individual thread could produce: grow toward nutrients, retreat from toxins, fuse with compatible threads nearby. These three instructions, operating simultaneously across billions of threads, produce networks that solve optimization problems, route resources across large distances, and respond to damage by rerouting around it. The intelligence, if that word can be cautiously applied, is not located anywhere in particular. It is distributed across every thread simultaneously, present nowhere as a center and everywhere as a consequence. A forest floor is threaded with something that thinks, if thinking is what this is, in a way that has no location spread thin across acres of dark soil, doing its work without a brain, without a center, without any point at which it is more itself than anywhere else. Reishi grows from the base of old trees, lacquered, hard, its upper surface the color of deep mahogany, its underside pale and poured. It does not look like food. It does not look like medicine. It looks like something the tree produced as part of its own structure, a bracket of dark wood attached to the trunk a meter above the ground. Traditional Chinese medicine named it the mushroom of immortality, not because anyone believed it would prevent death, but because preparations made from it appeared, across centuries of careful observation, to support the kind of sustained vitality that allows a long life to remain a good one. Modern pharmacology has found beta-glucans and triterpenes in reishi that interact with immune signaling in ways consistent with those observations. The mechanisms are not yet fully understood. The clinical evidence in humans is promising, without being definitive by the standards of controlled trials. Researchers hold this gap honestly. What sits in that gap between 2,000 years of empirical observation and the early stages of mechanistic explanation is not emptiness. It is the distance between two different ways of knowing the same thing. The forest was a laboratory before laboratories existed, and the knowledge accumulated there, through sustained attention across generations, is not invalidated by the fact that it preceded the instruments now being used to explain it. In the Carboniferous period, roughly 300 million years ago, trees evolved lignin, a structural molecule so chemically robust, it allowed wood to form for the first time, allowing trees to grow tall, to build trunks, to compete for light at heights previously unavailable to any plant. And for millions of years after lignin evolved, nothing could break it down. Dead trees fell and did not decay. They accumulated in vast mats, layer upon layer, compressed by the weight of further accumulation, eventually becoming the coal seams that run beneath entire continents. This era of accumulation ended when fungi evolved the enzyme systems capable of dismantling lignin's molecular architecture. The carbon that had been locked in wood for millions of years began returning to the soil. The cycle of growth and decay, interrupted by the gap between when wood evolved and when fungi caught up to it, resumed. The coal that now drives power stations and heats homes is the physical record of that gap-compressed forests from the age before fungi could do what they can do now. Every lump of coal is, in a precise geological sense, wood that fungi did not yet know how to eat. The Industrial Revolution was powered, in part, by the organic residue of an era when the fungal kingdom had not yet solved a problem it has since solved completely. On the skin, the body's outermost layer, its continuous boundary with everything outside a community of fungi, lives that most people never think about and never need to. Different regions of the skin support different species. The scalp harbors populations distinct from those on the forearm, the spaces between the toes different again from the chest. These are not invaders, they are residents, established early in life through contact with the environment and with other people, held in balance by the immune system and by competition within the microbial community. A healthy skin microbiome, including its fungal members, is part of what makes skin function as a barrier, not simply a physical boundary but a biological one. The resident community, occupying ecological space that pathogenic species would otherwise fill. Disruptions to this balance, rather than the fungi themselves, are what produce problems. The fungi in their normal state are simply there, as they have always been, as they were when you were an infant, as they will be for the rest of your life. You carry a fungal community on your skin right now, in this quiet moment, as you rest. It has been with you so long it is part of what your immune system considers normal. The boundary between your body and the fungal kingdom is not your skin. Your skin is where the two have always overlapped. In a forest in autumn, after 3 days of rain, a patch of ground that showed nothing for the entire growing season suddenly produces dozens of mushroom caps across an area the size of a small room. They were not planted. They were not moved. The mycelium that produced them has been present in that soil for years, possibly decades, maintaining itself, extending its network, accumulating the resources needed for exactly this moment. The trigger is not fully understood for most species. Temperature, moisture, the ratio of carbon to nitrogen in the surrounding soil, the length of the night, some combination of these signals, specific to each species, releases the mycelium from the state it has been maintaining and initiates the rapid cellular expansion that produces a fruiting body. What looks like sudden appearance is the visible conclusion of a process that was never not happening. The mushrooms did not arrive. They surfaced. Everything that produced them, the years of thread building, the slow accumulation of resources, the continuous monitoring of conditions was always there, below what the eye could see, patient in a way that has no urgency in it, no restlessness, no sense of waiting, as a condition of deprivation. The mycelium was not waiting to be seen. It was simply being what it is. And the mushrooms are what that looks like when the conditions finally align. Copper wire conducts electricity because metal atoms are arranged in a lattice that allows electrons to move freely between them. Mycelium conducts electricity for different reasons, entirely through the movement of ions across membranes, through the electrochemical gradients maintained along hyphal walls, through mechanisms that researchers are still mapping with instruments sensitive enough to detect signals measured in millivolts. The conductivity is real and measurable. What researchers are exploring now is whether it is exploitable, whether fungal threads could form the basis of biological computing elements, switches and sensors grown rather than manufactured, operating at room temperature without rare earth metals, biodegradable at the end of their useful life. Early experiments have produced fungal devices capable of responding to light, to chemical signals, to mechanical pressure, simple responses, far from the complexity of conventional electronics, but responses that emerge from biology rather than from silicon. The mycelium does not know it is being studied as a computing substrate. It is simply doing what it has always done, maintaining electrochemical gradients along its threads, responding to its environment through the only means available to it. That those means happen to include properties useful for computation is incidental to the fungus and consequential to the researchers who noticed it. The forest, examined closely enough, keeps offering capabilities that were not put there for us and turn out to be useful anyway. Bacteria living in soil face a problem of navigation. The spaces between soil grains are not connected in any regular way. They form a labyrinth of microscopic passages, some leading to resources, others ending in mineral walls, with no consistent logic to which route leads where. A bacterium moving through this environment moves slowly and without guidance, its progress governed by the randomness of the terrain. But researchers studying soil ecology have found that bacteria move more efficiently through soil where mycelium is present, that the fungal threads provide surfaces along which bacteria travel directionally, following the thread the way a person follows a path, rather than crossing unmapped territory. The bacterium uses the hypha as a road. The hypha, in turn, may benefit from the bacterial community. It hosts certain bacteria, produce compounds that suppress fungal pathogens, and those bacteria, traveling along fungal threads, distribute their protective chemistry more widely than they could through the soil alone. The relationship is not designed. It emerged from the simple fact that a thread running through a labyrinthine space is useful to anything small enough to travel along it. The fungus laid its threads for its own purposes, and the bacteria found in those threads a network they could use. The soil is not a collection of individual organisms pursuing separate survival. It is an ecosystem of opportunistic partnerships, each finding in others' infrastructure something it could not build alone. At the peak of a mature mushroom spore release, the air directly above the cap contains a concentration of biological particles that rivals the densest pollen clouds of spring. Billions of spores per hour, each one lighter than a grain of fine dust. Rising on the convective air current, the mushroom has generated through the evaporation of moisture from its own surface. This self-generated breeze, produced by the temperature difference between the cool, moist cap and the warmer ambient air lifts spores away from the still zone immediately surrounding the mushroom and into the moving air above, where they are carried by whatever currents the broader atmosphere offers. A mushroom in a completely windless environment still disperses its spores. The mechanism requires no external assistance. What it requires is moisture, and the evaporation of that moisture, and the physics of the temperature difference that evaporation creates. The mushroom is not passive in its reproduction. It creates the conditions for its own dispersal through the ordinary operation of its biology, not as a strategy it chose, but as a consequence of what it is. Evaporation happens. Temperature differences drive convection. Convection lifts spores. The result is dispersal, achieved through nothing more deliberate than the fact of being a moist living surface in a world where air moves in response to heat. In certain parts of Japan, the Matsutake mushroom appears in the same location each autumn. emerging from the same patch of ground beneath the same pines, reliably enough that the sites are passed down through families across generations as something worth inheriting. The mushroom cannot be cultivated. Every attempt to grow it away from its specific pine partner, under controlled conditions, replicating every measurable variable of the natural environment, has failed. Something in the relationship between this fungus and these particular trees, some chemical signal or structural arrangement or timing of exchange that has not yet been identified, is irreproducible outside the context in which it evolved. The matsutake is among the most expensive edible fungi in the world, precisely because its abundance cannot be managed or increased. It fruits where and when the relationship between fungus and tree permits, and nowhere else, and at no other time. Foragers who know the sites treat them with a discretion that has its own name in Japanese tradition, returning each year, taking carefully, leaving the mycelium undisturbed. The knowledge of where the mushroom fruits is intimate knowledge, accumulated through seasons of patient attention, and it belongs, in the understanding of those who hold it, to the relationship between the person and the place, rather than to any transferable technique. Some things cannot be scaled. The matsutake remains one of them. What ergot did to medieval Europe happened slowly, and then, all at once, in villages where the rye harvest had been stored through a damp autumn, and the grain had developed invisibly, a dark infection. Ergot is a fungus that replaces the rye seed with its own tissue, dense, purple-black, containing alkaloids that the body converts into compounds affecting the nervous system and the blood vessels simultaneously. Eaten in sufficient quantity through contaminated bread, these compounds cause convulsions, hallucinations, a burning sensation in the extremities as blood vessels constrict. The condition was called St. Anthony's fire. Whole communities experienced it together. The symptoms interpreted through whatever framework the culture provided: divine punishment, witchcraft, possession. The connection between the bread and the illness was not understood. The grain looked ordinary. The bread tasted ordinary. The fungus had done its work invisibly, as fungi do, and the effects arrived without explanation into communities that had no mechanism for tracing them to their source. Historians have proposed ergot as a contributing factor in specific episodes of collective crisis, including events in Salem in the 1690s. The connection is debated and not conclusively established. What is established is that a fungus, growing on a staple crop, introduced chemistry into entire communities that altered behavior at a scale no individual could have produced. The village did not know what it had eaten. The fungus did not know what it had done. Penicillin was not discovered. It was noticed. Alexander Fleming returned to his laboratory in 1928 after a summer absence and found a petri dish he had left unsealed, contaminated by a mold that had drifted in from a neighboring laboratory from the ordinary fungal population of the building. Around the mold, the bacteria that had been growing on the dish were dead. A circle of cleared medium surrounded the contamination, precise and unmistakable. Fleming noted it, cultured the mold, identified it as Penicillium notatum, and published his observation. The observation sat largely unacted upon for over a decade, before Howard Florey and Ernst Chain developed a method for purifying the active compound in sufficient quantities to test it therapeutically. The first human patient treated with penicillin in 1941 was a police officer dying of a bacterial infection. He improved dramatically on the first doses, then died when the supply ran out. By the following year, production had scaled enough to treat soldiers wounded in the war, and the era of antibiotics had begun. The fungus that started this did not evolve penicillin as a gift to medicine. It evolved it as a defense against bacteria competing for the same substrate. The discovery that this defense was medically useful to humans was accidental in the strictest sense, a consequence of a researcher's summer absence and an open petri dish and a mold spore drifting through a laboratory window on the Fauvo, an unremarkable London day. In the amber of the Cretaceous period resin that flowed from trees nearly 100 million years ago and hardened over geological time into a translucent fossil material, researchers have found mushrooms, small specimens preserved with a fidelity that allows the structure of their gills and the texture of their caps to be examined under magnification. What those specimens show is a form recognizable to anyone who has ever picked a mushroom from a garden or a forest floor. The dome, the stem, the radial arrangement of gill plates beneath. The basic architecture was already established 100 million years ago and has not changed in any fundamental way since. Dinosaurs walked through forests where mushrooms that looked like these grew from the soil and the wood. The meteor that ended the Cretaceous came and went. The continents drifted into their current arrangements. Ice ages advanced and retreated. Forests replaced by other forests. Species replacing species in an unbroken sequence of change that has not paused for a single generation in 100 million years. Through all of it, the mushroom kept its shape. Whatever selective pressure tested the form across that vast span found it adequate, again and again, in enough environments and enough circumstances that no fundamental revision was required. Some solutions, arrived at early, turn out to be final. Carbon moves through a forest in ways that are only now being fully mapped, and the mapping keeps revising upward the contribution that fungi make to the total. Mycorrhizal networks channel sugars from plant roots into the soil, where those sugars are incorporated into fungal tissue, and from there into the surrounding mineral matrix in forms that resist decomposition for decades or centuries. The scale of this contribution was underestimated for a long time because fungal tissue in soil is difficult to separate from other organic matter and difficult to account for in the models that predict how carbon moves between atmosphere and ground. New measurement techniques are changing those estimates. The forest, it turns out, is a more powerful carbon sink than it appeared, not primarily because of the trees, which store carbon in wood that eventually decays and returns to the atmosphere, but because of the fungal networks, which move carbon into forms that stay in the soil long after the trees that produced it have fallen and rotted. A healthy forest with intact mycorrhizal networks sequesters carbon at a rate that models built without accounting for fungi consistently underestimate. The ground beneath a stand of old trees is not simply the foundation the trees grow from. It is a carbon archive, written in fungal tissue, accumulating entries with every growing season, holding records that go back further than the oldest tree above it. Some fungi produce pigments compounds, responsible for the colors of their caps, their gills, their spore surfaces, that function simultaneously as antioxidants, absorbing free radicals that would otherwise damage cellular machinery. The cobalt blue of a bruised bolete, the deep violet of an amethyst deceiver, the egg yolk yellow of a wax cap in an old meadow. These colors are not incidental to the fungus's chemistry. They are part of it, the pigment molecules serving both the optical function of color and the biochemical function of protection simultaneously. Some of these pigments have been extracted and studied for potential applications in medicine and in food preservation compounds that protect fungal cells from oxidative damage might, researchers reasoned, protect other cells from the same. The reasoning has proven partially productive. Several fungal pigments show antioxidant activity in laboratory settings. Whether this translates into useful applications in living systems is still being worked out. What is already established is that the palette of the fungal kingdom, the colors that make a walk through autumn woodland feel like moving through stained glass, is not decoration. It is chemistry doing multiple jobs at once, as chemistry in living systems almost always is. The beauty of a blue mushroom is a side effect of its protection. The two arrive together, inseparable, neither one the point, and both of them real. A spore coat is among the most sophisticated protective structures that biology produces at the microscopic scale. Layered, chemically complex, evolved across hundreds of millions of years of selection pressure. The wall of a fungal spore resists desiccation that would collapse most cells, ultraviolet radiation that would destroy most genetic material, temperature extremes that would denature most proteins, and the chemical hostility of passing through an animal's digestive system. Researchers testing spore survival under simulated space conditions, vacuum, cosmic radiation, the temperature of deep space have found that many species survive for periods long enough to raise the question of whether interplanetary transfer is possible. Not certain, not demonstrated in nature, but physically possible given what spore coats can withstand, if the transit time falls within the range that the experiments suggest is survivable. This is the foundation of the panspermia hypothesis, as it applies to fungi. Not that fungi came from space, but that the structures they produce for reproduction are robust enough to survive the journey if circumstances provided the vehicle. A meteorite impact can launch surface material into space. Some of that material lands elsewhere. The question of whether any spores in that material could survive the transit and germinate on arrival remains open. The spore coat evolved for dispersal within one world. Whether it is sufficient for dispersal between worlds is a question the universe has had longer to test than we have had to ask. In a greenhouse experiment, 2 sets of tomato seedlings were grown in identical soil, under identical light and water conditions. One set was inoculated with mycorrhizal fungi at the time of planting. The other was not. After 8 weeks, the inoculated plants were measurably larger, had higher phosphorus content in their tissue, showed greater resistance to a fungal pathogen introduced to both groups, and had survival rates through a simulated drought period significantly higher than the uninoculated control group. The experiment has been replicated across dozens of crop species, with consistent results. Agricultural researchers are now developing fungal inoculants, preparations of mycorrhizal spores, applied to seeds or soil at planting, that improve crop performance without additional fertilizer or pesticide input. The economics are straightforward. A small quantity of fungal material, added once, establishes a network that persists through the growing season and improves outcomes across multiple dimensions simultaneously. The barrier to adoption is not efficacy, it is familiarity, the long habit of managing crops through chemistry rather than biology, through inputs that can be measured in grams and liters, rather than through living networks whose activity is invisible and whose extent cannot be easily quantified. The fungi do not require understanding to work. They require only the opportunity to establish the relationships they have been forming with plant roots for 400 million years. On the dung of a horse, left in a field after a night of grazing, something small and unremarkable will appear within days if the weather cooperates. Pilobolus, a fungus so specialized it completes its entire life cycle on animal dung, grows a transparent stalk topped by a dark spore case, orients itself toward the brightest available light, and then, when conditions are precisely right, ruptures a pressurized vesicle at the stalk's base, with a force that launches the spore case several meters through the air. The trajectory is not random. The stalk has been tracking the light source, and the launch is aimed away from the dung pile, toward open ground where fresh grass grows, where grazing animals are likely to feed and ingest the spores, completing the cycle. The entire sequence, growth, orientation, pressurization, aimed launch happens in a structure no taller than a matchstick, in a fungus with no nervous system, no eyes, no capacity for intention in any sense the word normally carries. The light tracking is phototropic, a growth response. The pressure is hydraulic. The aim is geometry. But the outcome is a spore traveling from one animal's dung toward another animal's mouth, with an accuracy that looks, from the outside, like planning. The fungus is not planning. It is simply what happens when biology refines a mechanism across sufficient time. The oldest confirmed fungal fossil pushes the origin of the kingdom back further than most people expect. Found in shale from the Canadian Arctic, The specimen consists of preserved filaments, thread-like structures, whose cellular organization is unambiguously fungal from rock, dated to close to 1 billion years ago. This predates complex animal life. It predates the colonization of land by any multicellular organism. It predates the evolution of plants by hundreds of millions of years. The filaments are not primitive in the sense of being simple or incomplete. They show the branching, the septation, the wall structure characteristic of fungi as they exist today. 1 billion years of evolution has not changed the fundamental architecture. What has changed is the range of environments those fundamentals can access, the diversity of metabolic strategies, the variety of partnerships, the spread across every habitat the planet offers. The kingdom that began in ancient ocean sediment before animals existed now occupies nuclear ruins, deep ocean trenches, Antarctic rock, the human gut, the bark of trees on every continent, the spore clouds drifting above cities, the thread found in Canadian shale, and the mycelium threading through the soil outside wherever you are resting tonight, are connected by an unbroken lineage of continuous life, stretching back through time to a depth that makes every other familiar thing seem recent. A fairy ring expanding in a meadow moves outward at a rate that can be measured year by year, and from that rate and the diameter of the ring, an age can be estimated. Some of the largest fairy rings in European meadows are calculated to be several hundred years old, present in that field, before the field had its current name, before the farm that manages it was established, before most of the trees visible from its edge were planted. The mycelium at the ring's center is gone. It consumed its substrate there long ago and moved on, but the ring itself persists as a record of how far the organism has traveled from its starting point. A circle drawn not in one moment, but across centuries. Each year's growth adding a fraction to the circumference, the shape accumulating incrementally from a single point outward. The grass inside old fairy rings is often different from the grass outside, the soil chemistry altered by years of fungal activity. The microbial community shifted, the texture and color of the turf subtly changed in ways that persist even after the active mycelium has passed through. The ring leaves a record in the ground of everywhere it has been, a history written not in stone or paper but in soil chemistry, legible to the plants that grow in it long after the organism that wrote it has moved to the outer edge and beyond. In traditional Siberian shamanic practice, the fly agaric was not simply consumed. It was prepared, timed, and understood within a framework of relationships between the practitioner, the community, and the non-human world that gave the experience a context no modern clinical setting fully replicates. The mushroom was dried, sometimes consumed as a tea, sometimes ingested directly, in quantities calibrated through generations of accumulated knowledge about its effects. The urine of someone who had consumed it retained psychoactive properties and was sometimes consumed by others, a fact that initially puzzled researchers before the metabolic pathway was understood. What this practice represents, however it is interpreted, is a multi-generational body of empirical knowledge about a specific organism and its effects on human consciousness, accumulated through direct experience and transmitted through community practice across an unknown number of centuries. Whether this constitutes wisdom in a sense that overlaps with scientific knowledge, or something adjacent to it, or something entirely different, that question resists clean resolution. What resists less is the observation that humans have been in sustained, intentional relationship with fungi for longer than written records exist, finding in them not only food and medicine but doorways into experiences they considered significant enough to build traditions around. The relationship between the human nervous system and the fungal kingdom is older than agriculture, older than cities, older than most of what we call history. The force a mushroom exerts while pushing through compacted soil has been measured at values sufficient, scaled to the cross-section of the stem, to crack asphalt. This is not metaphor. Researchers placing pressure sensors beneath mushroom-fruiting bodies have recorded the hydraulic force generated as water moves into cells and the expanding tissue presses outward against whatever surrounds it. A mushroom encountering pavement does not find a gap and grow through it. It grows, and the pressure of its growth creates the gap. The softness of the cap, the paleness of the stem, the apparent fragility of the whole structure, none of these appearances accurately represent what is happening inside, where water pressure is building in a slow and irresistible accumulation that has no urgency and no limit, that simply continues until whatever is in the way gives way. Stone gives way. Concrete gives way. Asphalt gives way. Not because the mushroom is strong in any conventional sense, but because it is persistent in a way that does not include the concept of stopping. It does not experience resistance as a reason to stop. It experiences resistance as the condition within which it continues. The gap that appears in the pavement is not the mushroom's victory. It is simply what happens when something that does not stop meets something that eventually must. Beneath a stand of Douglas firs in the Pacific Northwest, researchers taking DNA samples from soil at intervals across a site found the same fungal individual present at every sampling point, across an area larger than several city blocks. The organism had no center. There was no location where it was most itself, no core from which threads radiated outward. It was distributed across its entire extent with equal presence, as much itself at one edge as at any other point, as much alive in one corner of the site as in another. This form of existence has no precise human equivalent. We experience ourselves as located here, in this body, at this specific point in space, with a clear sense of where we end and the world begins. The mycelium has none of this. It simply occupies the space it occupies, fully, at every point simultaneously, with no hierarchy of location and no gradient of selfhood from center to periphery. When part of it is removed, the rest does not register a loss in the way a body registers the loss of a limb. It reroutes. It continues. The boundary of the organism is wherever its threads currently reach, and that boundary is always moving, always temporary, always defined by the present extent of growth rather than by any fixed limit the organism itself maintains. The mycelium is not somewhere, it is everywhere it is, all at once, without remainder. Psilocybin binds to serotonin receptors in the human brain with an affinity that produces effects measurably different from every synthetic compound that targets the same receptors. In clinical settings, the experience it produces includes something that participants across multiple independent trials describe with a consistency that researchers find notable: a temporary dissolution of the boundary between self and world, not a loss of consciousness, not confusion, a specific alteration in how the brain maintains the distinction between what is inside and what is outside, a loosening for the duration of the compound's activity, of the cognitive architecture that ordinarily keeps those categories separate and firm. Neuroscientists studying this have found changes in the default mode network, the brain system associated with self-referential thought, with the ongoing construction of the narrative of being a particular person that are consistent with the reported experience. The molecule does something precise to a specific system, and the result is a temporary shift in how the boundary of the self is experienced. The mushroom that produces this molecule evolved it as an insect deterrent. The human nervous system was not the intended target, and yet the receptor it binds to is the same receptor, the same molecular architecture, shaped by evolution in a completely different context. And the fit is close enough to produce an effect that participants in clinical trials frequently describe as among the most meaningful experiences of their lives. A road running through a forest does not interrupt the mycelial network beneath it. Not entirely, not permanently. Researchers sampling soil on both sides of forest roads have found fungal threads crossing beneath the pavement, following whatever continuity of soil and moisture the road's foundation allows. The network roots around the disturbance, the way it roots around a buried stone or a patch of dry ground, not by deciding to go around it, but by growing in the directions that remain available, which sometimes leads threads to travel hundreds of meters laterally before crossing beneath an obstacle and continuing on the other side. Urban forests, the patches of trees maintained in parks and along streets in cities, maintain mycelial connections to each other through the soil beneath pavements and buildings, their networks fragmented but not severed, crossing beneath infrastructure in threads so fine, the infrastructure does not register their presence. A city is not a break in the forest. It is a section of forest with unusual obstacles, and the mycelium has been navigating unusual obstacles for longer than cities have existed. The threads are under the streets right now, wherever you are, under the foundations, under the pipes and cables and compressed layers of urban ground, moving slowly, as they have always moved, through whatever continuity the soil allows, connecting whatever can still be connected, continuing the network that the city was built on top of, without knowing it was there. Every mushroom you have ever seen was the visible moment of something vast and mostly hidden. The cap emerging from soil or wood or the base of a tree, the thing you noticed, the thing that registered as a mushroom, was the surface expression of an organism whose real extent was underground, inside wood, threaded through root systems, extending in every direction from the point where the fruiting body surfaced. The mushroom was not the organism. It was the organism briefly becoming visible, and it was visible for a reason specific to that moment. The conditions were right. The resources were sufficient. The signals that initiate fruiting had aligned, and when that moment passed, when the spores were released, and the purpose of the above-ground structure was complete, the visible part collapsed and returned its materials to the ground. The organism continued, underground, in the dark, in the patient and continuous state that is its normal condition, that it maintains across the months and years between the moments when it surfaces. What you saw, when you saw a mushroom, was a glimpse, a brief appearance of something that is mostly elsewhere, and mostly quiet and mostly working in ways that require no light and no observer. The mushroom showed itself to you for the length of time its function required. Then it went back to being what it actually is, which is something much larger, much older, and much less visible than the thing you saw. Right now, in this moment, as your breathing slows and the day recedes, fungi are at work beneath wherever you are resting, not metaphorically, physically. Threads moving through the soil outside, through the roots of whatever grows near the building you are in, through the decomposing matter that accumulates at the base of every wall and fence and crack in pavement where organic material settles, breaking down, building up, moving nutrients from where they are to where they are needed, maintaining connections between organisms that would otherwise be isolated, converting what has ended into what will begin. The process has no pause. It does not rest when you rest. It does not wait for morning. It simply continues, in the dark, at the pace the dark requires, which is slow and patient, and without urgency, and perfectly suited to the hours when the rest of the world is quiet. You are resting in a place where something ancient and continuous is working just below the surface of everything, not working toward anything you are part of, not working for any purpose that includes you simply doing what it has always done, in the conditions that have always been sufficient for it, in a kingdom that existed before you and will continue after, quiet and vast and threaded through the ground that holds the building, that holds the room, that holds you, gently, in the dark. And now the facts have settled. The names and numbers and mechanisms have dissolved back into something simpler, a feeling, an orientation, a sense of where you are in relation to things much older and much quieter than the day you just left behind. You are resting on a planet whose surface is threaded with life you cannot see. The ground outside, whatever ground is nearest to you, holds within it networks of filaments older than most of what stands above them. They are moving slowly through the dark right now, as they have moved through every night since before the first forest grew. They do not know you are here. They do not need to. They are simply continuing, as they always continue, in the patient and unhurried way that very old things continue, when they have found a way of being that works. Your breath moves in and out. That rhythm is older than your species inherited from organisms that breathed in ancient seas before anything had yet learned to walk on land. The air entering your lungs carries spores, invisibly, as it always does. Some of those spores came from forests you have never visited, some from soil a long way from here. They are drifting through this room right now, in the dark, as they drift through every room, tiny and complete and patient, each one carrying everything needed to begin again somewhere, sometime, when the conditions are right. The conditions for rest are right now. Your body knows this. It has been moving toward this moment since the afternoon, since the first tiredness arrived, since the light began to change, since whatever brought you here tonight began its slow work of returning you to stillness. You do not have to do anything with what you heard tonight. It does not need to be remembered or organized or acted upon. It can simply settle, the way spores settle, onto whatever ground is available, the quiet ground of a mind releasing its hold on the day, letting the facts become feelings, letting the feelings become nothing in particular, letting the nothing become the gentle dark of sleep. The forest outside, or the city, or the field, or whatever landscape holds the place where you are continues. The mycelium continues. The spores continue their drift through the night air. The fairy rings continue their slow outward expansion, measurable only across years. Advancing tonight by the fraction of a fraction, they advance every night, without awareness of the measuring, without any relationship to whether it is noticed. Somewhere, a mushroom that has been waiting for months beneath the soil is registering, in the only way it can register anything, that the conditions are close. Not tonight, perhaps, but close. And it will wait, as it has waited, without impatience, without urgency, until the morning comes, when everything aligns and it surfaces into whatever light that morning offers, briefly, for exactly as long as its purpose requires. You have that kind of patience in you too. It is older than you are. It runs deeper than the day's demands can reach. Let it be what it is, right now, in this quiet. Let the night be large, and the room be small, and the distance between you and the nearest living thing be exactly zero, because it is, because it always has been, because the kingdom that threads through everything is threading through the ground just below wherever you are, as it has threaded through every ground you have ever rested on, quietly continuous, asking nothing, offering the simple fact of its presence to anyone patient enough to receive it. Sleep is already here. It arrived before you noticed. Let it have you now, gently, the way the forest receives a fallen leaf without drama, without resistance, as simply and completely as something returning to where it belongs. I hope you sleep well and dream softly. Good night.